Good morning or afternoon, everyone. On behalf of CIRM, I would like to welcome everybody to the CNS Consortium Workshop in what we hope will be a very informative 2 days of talks and discussions that will inform CIRM in its mission and moving forward. In the next 4 slides, I'll provide a quick overview of the history leading to our mission and the goals, and to this workshop and the goals that we have set for this workshop. Next. So the CNS workshop is framed within the new and revised mission statement, reflecting the new era that CIRM embarked since the passage of Prop 14. And the changes to our mission statement reflect how the science, technology, and the needs of our society have evolved in the past few years. And specifically, the use of the verb accelerate, as in accelerating world-class science, is fundamental to CIRM's identity and value proposition and what we are trying to do today. CIRM accelerates through its funding model, through partnerships, and also through promoting a culture of collaboration and team science, which is partly why we are here. CIRM's commitment is to developing a systematic approach that fosters team science. Next. The vision of today's workshop has been informed by multiple layers of stakeholder discussions and input that started even prior to the passage of Prop 14 over the past 2 years. Next. Starting in 2019 with the brainstorming neurodegeneration workshop, CIRM received broad stakeholder input about the benefits and also the bottlenecks of developing a consortium approach where genomics and big data, novel stem cell models, and patient data could be collectively leveraged to advance the field of neurodegenerative research in a collaborative manner. Next. In 2020, just before the passage of Prop 14, and in order to better understand the status of bottlenecks of knowledge networks that could leverage this data sharing, we actually brought together a panel of experts in the context of our yearly grantee meeting. We were encouraged to learn that the approach for promoting knowledge sharing was right on target, and that the panelists suggested that CIRM had a great opportunity to promote a paradigm shift in this area. Next. In the early 2021, last year, immediately after the passage of Prop 14, which was November 2020, and building upon our previous conversations, we formed a strategic scientific advisory panel that was comprised of distinguished group of national and international scientists in the stem cell field. And once again, during that meeting, we were advised to expand shareable resources, especially in the context of stem cell modeling, and bring more attention to complex diseases such as neurodegenerative and neuropsychiatric disorders, and facilitate a knowledge sharing. Next. In the mid of 2021, as we were forming our new strategic plan, based on all this input, we pressure tested our paradigm shift vision in a town hall meeting and further gathered feedback from California stakeholders about their needs. Again, all arrows pointed to shared resources as data as critical elements for accelerating research. Next. Finally, in late 2021, just before the launch of our strategic plan, we organized an advisory committee meeting, and this committee was gathered to advise CIRM on ways to facilitate collaborative knowledge sharing. And here, during this meeting that happened last September, we explored various models to leverage and or also generate a data infrastructure in which CIRM-funded data could be managed and shared. And the main outcome of this meeting was a recommendation to organize this workshop to test the feasibility of this collaborative approach. Next. Finally, in December of 2021, as many of you know, and you've seen it because it was part of the materials that we sent through the program of this meeting, paralleling the input we had received from experts and stakeholders, we launched our 5-year strategic plan with the goal of advancing world-class science by promoting a culture of collaboration. The how to is materialized, the how to promote and advance world-class science is materialized in today's workshop, in which we will be digging into more specifics around how to enable this collaborative ecosystem. Next. And here we are. The first of the three main themes of the strategic plan is to advance world-class science by leveraging collective scientific knowledge to inspire collaborative research. And as articulated in our new strategic plan, CIRM is committed to continue funding high-risk, high-reward research projects through our existing pillar programs, the discovery, the translational, and the clinical, and to promote and the networks and fosters shared usage of the specialized technologies through competency hubs. The approach that we are proposing, that has been proposed to the strategic plan, is to build an infrastructure that organizes and democratizes data through the development of next generation technology competency hubs, and second, the building of knowledge networks to facilitate data sharing and to foster a culture of open science. The goal of this two-day workshop is to gather feedback that will inform CIRM about potential opportunities for best approaches to achieving this goal. While the discussions at the workshop will center on applications to the central nervous system as a use case, the outcomes will be implemented broadly across cell types, organs, and diseases. The data infrastructure is intended to be a global resource to facilitate data sharing and foster a culture of open science for all certainties and ultimately also the world to collaborate with the world. Next, at a high level, we have structured the next 2 days of discussions around two main themes. Day one will focus the focus of day one is to gather feedback around opportunities to share resources and promote collaborative research in California, and specifically through the creation of competency hubs. Ute Grishammer will provide a more comprehensive overview in a few minutes. For day two, the focus is going to be around finding the best approaches to promoting data sharing and whether this is a role for CIRM in supporting this or being part of it. Next slide. A bit about the logistics of this workshop. There's a splash slide that will be projected at regular intervals with the instructions and logistics of the workshop. But quickly, I move these slides. Can you go to the next one? And then we'll go back to this one. This is a public workshop with an audience in the webinar part of the meeting that will be in listening only mode. For questions for this audience, please use the Q&A. CIRM is managing the questions and will relay to moderators of the discussions. Also, all questions will be recorded and be subject to an FAQ later on. Not all questions will be responded. The speakers for both days are stakeholder experts, funders, and users of the types of infrastructure, tools, and resources that we will be discussing. And the discussants are additional experts and board members, as well as CIRM staff. The slide previous. This is a quick overview of the workshop attendees. The total attendee is now over 150 with representatives over 50 institutions, including the types of organizations that you see in this slide. And in the interest of time, we have provided a program that contains details and bios of all the speakers and discussants. Therefore, introductions to speakers will be brief. I would like now to introduce Ute Grishammer, who is a Senior Science Officer in the Scientific Programs team at the California Institute for Regenerative Medicine, and who will be the Master of Ceremonies for today's session. Ute's name is also very familiar to many of you, as she returned to CIRM last year, and she was actually previously at CIRM from 2007 to 2015, and managed the programs that created both the CIRM Genomics initiatives and the IPSR repository, which we will hear a lot about. Ute, thank you.
Thank you, Rosa. And I hope everybody can hear me. Good morning. And welcome to all attendees and participants. As Rosa said, I'll be the MC for today's part of the workshop. There I am. Thank you. And I look very much forward to the presentations this morning and then the discussions this afternoon. Next slide. So the focus of today is to gather feedback around opportunities to share resources and promote collaborative research in California through the creation of competency hubs. We have structured today's part of the workshop into 2 parts. Session 1 this morning provides an overview of previous CIRM-funded research resources. And then session 2 this afternoon will be a discussion about how to share resources and promote collaborative research going forward. In case you're wondering what we mean by Competency Hub, I will provide an explanation at the beginning of session 2 when we start talking about feedback for potential future opportunities. Next slide. For now, we'll focus on previous CIRM-funded research resources to provide a historical context and information to consider this afternoon when we will be thinking about future opportunities and whether to leverage these resources, and if so, how. In the next few minutes, I'll provide an overview of the CIRM Shared Laboratories Program, the CIRM Human-Induced Pluripotent Stem Cell Repository, and the CIRM Stem Cell Genomics Centers of Excellence. The presentations that then will follow once I'm done with my overview will be specific examples of how these resources have been implemented and used. Next slide. So I'll start with the Shared Labs program. The objectives were to provide dedicated safe harbor research space, specialized instrumentation, cell lines, culture materials to be used by multiple investigators at grantee institutions and neighboring research institutions, and in particular for the use of human embryonic stem cell lines that fell outside the federal guidelines back then. And so this was a upcoming field of human embryonic stem cell research that CIRM wanted to support by providing these opportunities for shared laboratories. Another objective was to provide training and instruction in stem cell culture and technologies through formally offered courses and one-on-one customized instruction. The funded shared labs were operating until 2016. Many are still ongoing, and we'll hear about one of them from Dave Schaffer in a moment. 17 shared labs awards were made to academic and nonprofit research institutions. They were equipped to grow and maintain human embryonic stem cells and later also human-induced pluripotent stem cells. And they developed, many of them developed and implemented specialized services or tools such as derivation of human pluripotent stem cell lines, imaging facilities, omics services, vector production, and many others. In addition to training for the use of the shared labs, 6 shared labs were also funded to develop and provide an advanced techniques course, which was offered, for instance, to students in some of CIRM's training programs. As I mentioned, Dave Schaffer will speak about the UC Berkeley experience. Next slide. The second research resource I'd like to highlight is the CIRM Human-Induced Pluripotent Stem Cell Repository. The objective of that program was to create a comprehensive collection of research-grade iPS cell lines for reliable distribution worldwide for modeling of prevalent genetically complex diseases. The funded awards led to the creation of a centralized iPS cell bank that contains iPS cell lines from over 2,000 unique donors. These iPS cell lines were created with a uniform production method. They're consented, appropriate for intended use, research use, and demographic, medical, and or diagnostic information from each donor is included in this collection. Next slide. Here I'm showing an overview of the diseases that are covered in the CIRM iPS cell repository, the patient tissue samples that were used for the creation of the iPS cell lines were collected by clinician scientists from the institutions highlighted on the right. Most iPS cell lines represent indeed prevalent genetically complex diseases. And there's also a representation of some rare diseases in this collection. controlled samples from healthy, unaffected donors were also included. The IPS lines were derived by Cellular Dynamics International, and they are now distributed by Fujifilm Cellular Dynamics, FCDI. So we have 3 short talks coming up that illustrate the use of this resource. Next slide. So my last 2 slides before we go into this morning's talks provide an overview of CIRM's Genomics initiative. The objective was to enable application of cutting-edge genomics approaches to substantive problems of human stem cell biology. The funded awards led to the creation of the Center of Excellence for Stem Cell Genomics, which was a 7-institution collaboration that included a data coordination and management center. The Genomics Center participants were funded to conduct their own research projects, and they provided genomics support to an additional 14 stem cell projects at 8 institutions. Next slide. The combined efforts of the research projects and the Data Coordination and Management Center led to new genomics-based stem cell knowledge with over 40 publications as of now. I'd like to remind you that single-cell omics approaches were emerging as a major player at the time of establishing the Stem Cell Genomics Center. And this initiative contributed to bringing this data-intensive and technically challenging approach to stem cell labs in California. Much of the omics data generated by the Genomics Center that's illustrated on the right was single-cell data. The Stem Cell Genomics Center also developed data analysis tools, including the cell browser to visualize single-cell data. Aparna Baduri and Max Häusler will talk about that in a moment. The center also created reporting standards for metadata, an important aspect of creating data collection by the Data Coordination and Management Center. The guidelines, for instance, for reporting single-cell RNA-seq experiments have been published. And the center also developed guidelines for minimal information about a stem cell experiment, which is available on the Stem Cell Hub web portal. And then finally, a data warehouse was created by the Data Coordination and Management Center. They ingested, curated, and stored 84 terabytes of data that was generated by the projects that were part of the Center of Excellence. This data is available through the web portal called the Stem Cell Hub. And some of this data is public, but most is made accessible to authorized users. Okay, next slide. So we will now have 5 short talks illustrating some of the outcomes from these CIRM Proposition 71 initiatives. As also mentioned, the introductions of speakers will be very brief. Please refer to the workshop program for more information about the speakers. And if all goes to plan, there should be time for a question or 2 after each talk. For the live participants, as a reminder, you can raise your Zoom hand, and the attendees for the webinar can post questions in Q&A. So now I would like to hand it over to Dave Schaffer, which I haven't seen earlier, but I hope is present by now.
I'm here, yes. How are you doing?
Yay. Hi, Dave. All right. Take it away, Dave.
Great.
Sorry, Dave, before I did want to say just 5 seconds, to introduce you just very briefly. Dave Schaffer is a professor at UC Berkeley. He's the director of UC Berkeley's Stem Cell Center, and he was a director of the CIRM-funded shared lab at UC Berkeley.
Great, thanks so much. Just one slight update. I was director of the Berkeley Stem Cell Center for 10 years. Berkeley has a policy that we're term limited at 10 years. So I stepped over into being director of QB3 about a year ago or so. However, I still am director of this facility. So just to dive into a little bit more detail with what Jutta mentioned on what was RFA 0701, the CIRM Shared Research Laboratory Grants and Stem Cell Techniques course, I'd like to dive in a little bit of detail into how Berkeley has benefited from this program. Next slide, please. So I'm not going to go into this in much detail because Jutta already, I think, really highlighted this very well, but they had shared the objectives for this program. And primary amongst these is the fact that, of course, at that time, there were a lot of non-registry, non-government authorized cell lines, human embryonic stem cell lines. And this facility had the goal of providing square footage that was not, did not have a connection to federal or NIH funding, such that investigators would be free to be able to utilize this square footage for the culture of human embryonic stem cell lines outside the federal guidelines, or for other related research. Next slide as well. And in addition, as she mentioned as well, there were 6 awards for funding stem cell techniques courses. And the closest to us was UCSF. So the goal for this is that culturing human embryonic stem cells in high quality was at that time, a very specialized knowledge set. So there was an interest in developing courses to enable people from labs, graduate students, postdocs, or faculty to get access to that knowledge. The mechanism provided funding for 3 overall destinations. One was the capital projects to actually build up the lab space and to purchase equipment. Second was the initial installment of that RFA provided for 3 years of annual operations budget. And then the third was is the stem cell course, of course. And then in addition, there was actually an amendment onto this original RFA that had the effect of extending the operating budget for an additional 5 years, which was quite generous. So the original RFA was intended to invest around $50 million into what ultimately became 17 awards, as Jutta mentioned. So we're talking on the order of $3 million per campus or per facility. So this was sufficient to really outfoot several hundred square feet of space to be able to be dedicated for this purpose. And then with the extension of the operating budget, ultimately around $68.5 million was invested by CIRM. Next slide. The way that we implemented it on the Berkeley campus was that we had a joint facility that was used by Berkeley as well as by Children's Hospital Oakland Research Institute. Corey is just several miles down the road from Berkeley in Oakland. So this was intended to benefit the entire East Bay. And we called it the CIRM HESC Shared Research Facility. So the director that I hired originally back in around 2008 to run this facility is Dr. Mary West. And we are fortunate that we've retained her throughout this entire time. So Mary is still director of this facility, and I'm remaining the academic supervisor of the facility. It is still well used. I'll talk about how we've adapted to the sunsetting of CIRM a little bit later on. But just to give you a sense, last year, it was used by 43 research groups, PIs, and by 100 researchers from those research groups. The majority of it is in one building, Stanley Hall, which is also the headquarters for QB3. So it's the building that I run. And but a small outfit, specifically the part that contains the cell sorter, we decided made sense to actually put into the main campus flow cytometry core, since there was already a lot of expertise there, of course, to operate those kinds of instruments. Next slide, please. The main space had the goal of providing cell culture facility. That was one of the main goals. And so we had, three cell culture hoods, TC microscope, cryo storage for cells. And this was initially used for non-registry HESC lines, and then like the UCSF lines. So we're using HSF6 out of Susan Fisher's group. And, with the transition to the Obama administration, which we all know led to a dramatic increase in the number of federal registry lines, at that stage, it was not as imperative to have space for the culture of non-registry lines, but it ended up being transitioned for another purpose, which is the fact that on the Berkeley campus, we, in addition to having a large number of biology investigators, we also had physical sciences and engineering labs that progressively began to move into the stem cell area. And their main labs lacked cell culture space often. And so this ended up being a kind of a haven for them to be able to do their work and get access to high quality cell culture space. We didn't, as I mentioned, at Berkeley have one of the stem cell technologies courses, but many of us attended UCSF's course across the Bay. Again, thanks to Susan Fisher. Next slide. I'm not going to go through these in great detail since a lot of these instruments are workhorses that are going to be familiar to many of you. But we got a Becton Dickinson influx cell sorter, which was only the second sorter that the campus had at that time. So this got a large amount of volume. It was well outfitted with a lot of laser lines, which of course immunologists love. And this has gotten a lot of heavy use. We still have this instrument on the campus. Next slide. The other major piece of instrumentation that we got was a 2-photon and visible confocal fluorescence microscope. And this has enabled large amount of both visible fluorescence imaging, as well as some more complex techniques related to multiphoton, including multiphoton excitation, bleaching, ablations within cells. And I'll show you a couple examples of that. Next slide. So it enabled, kind of conventional 2D imaging like this. This is an image of midbrain dopaminergic neurons where the red is tyrosine hydroxylase. This is a 2-dimensional culture of human embryonic stem cells. Next slide. Of course, and if you could hit the next advancement on the animation. Of course, in the past 10 years, there's been an increased interest in culturing organoids. So these get, of course, several hundred microns in diameter, and it becomes imperative to use a confocal fluorescence microscope. So we've seen increased use of this system for imaging in 3D organoids. Next slide. And there are also some rather advanced users who've been harnessing this system, such as Marquita Landry's group, where Marquita has developed some carbon nanotube-based sensors of neurotransmitters, where these sensors actually fluoresce in the infrared region. And as a result, the infrared laser used typically for multiphoton excitation serves as a radiation source for single photon excitation of these infrared sensors, such that she can actually image the release of dopamine within, for example, brain slices. So she's been using this for a number of years to be able to analyze both healthy as well as diseased brain sections, both under degenerating conditions as well as upon the implantation of human embryonic stem cell derived replacement cells. Next slide. Another workhorse that was unanticipated. Those are the 2 major pieces of equipment that we got into the facility, but the facility has been evergreened through the addition of additional equipment from other sources. So this is actually a piece of equipment that we had got due to a faculty retention. Somebody who was going to get, of course, recruited to another university, asked for this equipment in order to stay at Cal, and quote unquote donated this equipment for use within the facility. So this is a high content fluorescence imager, which has seen a huge amount of use actually. Next slide. So one example is that it's been harnessed by a number of groups for high throughput screening analysis of stem cell differentiation. This is a slide where investigators used a fluidic system to be able to put 532 microcultures onto the slide. And then where each one is exposed to different culture conditions, different culture media, different matrix conditions for growth. Next slide. So that the investigators could get very high content data by using this with the fluorescence imager to be able to quantify the fraction of cells in each one of these 532 microcultures that had differentiated into a particular target lineage, in this case, a liganetocyte precursors. And this enabled optimization of conditions, both the number of agonists and identity of agonists that were used, the concentrations used, as well as the timing of addition in order to optimize differentiation into that target lineage. Next slide. We've also, there's a campus fund where a variety of core facilities can apply each year to be able to get the proceeds of a campus endowment for evergreening of equipment. This is, much smaller equipment, but, we're able to get a chamber where you can culture under a variety of different gas conditions, including hypoxia conditions. Next slide. We're able to obtain a, real-time fluorescence imaging system where you can place cells into an incubator under a microscope so that you can gather movies longitudinally for hours or even days as cells are being cultured in multiple channels of fluorescence. Next slide. And we're also able to get an analytical flow cytometer. I believe this was as a result of investigators Howard Hughes equipment money. Next slide. And finally, a small piece of equipment that has also had a big impact is we're able to get a multiplex bead reader for high content immunoassays. This is from Bio-Rad. Next slide. The current status of this is that we feel quite good that the Berkeley and Corey campuses strongly benefited from the goals of this RFA. So it was heavily used for a number of years and continues to be used for both registry as well as non-registry human embryonic stem cell culture. Initially, it was non-registry. I don't believe there's anybody on the campus still working with non-registry lines. We've been able to evergreen it through the addition of smaller pieces of equipment just to keep things somewhat up to date. However, as we all know, core facility expenses, and the two major ones are to pay for high quality staff, as well as for service contracts to keep the equipment in good shape. The expenses are higher than recharge income. And we have both campus sources as well as outside sources of recharge, where the outside sources we charge at a higher level than campus users. But even so, tapping into all these possible sources of equipment, the recharge is substantially lower than the expenses. So such facilities need to be subsidized by the campus or from other sources if the rates are to stay reasonable. Otherwise, the rates, of course, go up, and it becomes, investigators get more incentivized to start moving to other places to get their work done, including CROs. So CIRM funding sunset, or the last year where funding was given out for this mechanism, was 2015, which extended into 2016 for 1 year of funding. And then after that, QB3, which is the California Institute for Quantitative Biosciences, QB3 Berkeley took on the subsidy for the facility. Thankfully, they had some capacity within their budget to do so. And apologies to CIRM, but we decided that we wanted to, in order to keep the facility alive, broaden the name beyond stem cells, since investigators on the campus were, under the misconception that the facility was could only be used for stem cells. So we rebranded it as the QB3 Cell and Tissue Analysis Facility to be able to capture larger numbers of users and increase our recharge so that we could stay alive. So that was done in 2018. So our current status is that we have one staff person, Mary, part-time. Her time is split with another facility on the campus. She can't devote all of her time to the facility, and so she's offloaded onto a number of super users, a number of groups that use pieces of equipment at a high amount. So this has led to some drop in service because they can't respond instantaneously, and there's a lot of turnover within those groups. The two large pieces of equipment, the flow cytometer and the multiphoton confocal, are still around but are showing their age. And the multiphoton, due to the fact that a lot of the components are actually no longer manufactured by Bruker, its utility is just about at an end because we can't get replacement parts for it. But the good news is that this facility is still up and running and is still benefiting investigators across the campus as well as within the East Bay as a whole. So thanks so much for the time to describe this, and I'd be happy to take any questions.
Thank you, Dave. This was a fabulous introduction, to the details of a shared lab, and will be definitely a basis for discussion later this afternoon. As part of that discussion, we'll talk about, what does it take to incentivize and run shared resources labs, and we'll be talking about the next generation of that, but this gives us some fodder for thought as to what it takes for a shared resource lab like this particular one to maintain and keep running. So thank you for that. I'll see if there are any specific questions coming in from the participants. I think if somebody raised their hand, they would be coming to the forefront here of my little list of squares. And seeing none, I'll suggest, so, Dave, you'll be participating later in the discussion, and we'll come back to some of the things you talked about. Thank you so much. Our next talk is the first of 3 talks that represent uses of the human induced pluripotent stem cell repository that CIRM created. And there have been some just really fabulous examples of using this resource especially considering the fact that it's a large resource, and we have some use of that really takes advantage of the fact that we have many cell lines that were derived in a similar fashion and can be used in large-scale studies. So the next talk is actually a recording because the 2 speakers unfortunately cannot join us today. The speakers are both from the Broad Institute of MIT and Harvard. Ralda Nemi is a group leader at the Stanley Center for Psychiatric Research at the Broad, and Dia Ghosh is a computational biologist. And they'll explain how they were collaborating with CIRM in obtaining a lot of these cell lines and the human population genetic studies they've done with them. So if you guys could start that recording.
Thank you very much for giving us this opportunity to share with you some work that we've been pursuing on exploring the effects of genetic variation on cellular traits using a large collection of induced pluripotent stem cell lines and population scale cellular models. Now, as many of you may be well aware, accessible collection of high quality induced pluripotent lines for modeling disease and drug discovery. To this end, blood and skin cells were collected from 4 institutions and reprogrammed into iPSC lines by Fujifilm Cellular Dynamics. And at present, the repository houses cell lines derived from over 2,500 healthy donors and patients spanning a variety of neurological, metabolic, and cardiovascular disorders. Now, importantly, as part of standard QC, each distributable cell line has both array-based genotype data and is linked to demographic and clinical information reported by the donor. Recognizing the utility of this large collection of stem cells for studying how genetic variation might shape molecular and cellular phenotypes, our group at the Stanley Center and Harvard embarked on a close collaboration with CIRM and CBI to acquire large numbers of iPSC lines selected using the existing genotype and metadata. To add to this resource, we in turn generated whole genome sequences of all cell lines and have made this data publicly accessible through the Anvil database. Now, an important prerequisite for onboarding the CIRM iPSCs was to identify lines that would be maximally beneficial for our investigations. So to this end, we analyzed the array-based genotype data to infer genetic relatedness, ancestry, and sex. And this allowed us to weed out any sample duplicates or mismatches by confirming against donor metadata, resulting in a final set of 267 distributable lines from unique donors. The data revealed the presence of 11 trios and hundreds of parent-child duos, which was largely consistent with self-reported relationships. Since such panels of related individuals can be exploited to assess heritability of cellular phenotypes, we use this criteria to select our initial tranche of cell lines. Furthermore, we observed that the CIRM collection, in contrast to existing iPSC cohorts, is actually quite genetically diverse. In fact, while a majority of donors were of European descent, several individuals were from Asian, African American, and Latino or Native American ancestry, underscoring the value of this resource for investigating population-specific genetic variation and fine mapping studies. Now, in addition to inferring genetic background and relatedness, donor genotypes can also be used to select cell lines based on estimates of polygenic risk score for diseases of interest. As we considered experimental designs to ascertain how genetic backgrounds of donors shape disease risk in psychiatric disorders, we estimated the polygenic burden for schizophrenia across all of the CIRM donors. The first iteration of this analysis, in fact, across 500 or so donors, was leveraged in an NIMH collaboration with Kristen Brennet's group to nominate cell lines with high versus low risk scores for downstream assays. While the predictive power of such scores may be limited, we anticipate that prioritizing cell lines with high and low risk scores could be relevant in genetic perturbation studies and can actually unveil important clues about disease-relevant pathways or mechanisms. Now, beyond estimating polygenic risk score, we also harnessed the CIRM donor genotypes to impute common variants and haplotypes of interest for purposes of cell line selection. Finally, we leveraged this data to run concordance checks on all cell lines acquired prior to whole genome sequencing. And to date, we have successfully onboarded nearly 450 CIRM iPSC lines. For the rest of this talk, I will now focus on how we're using this resource to map the effect of genetic variation and cellular traits. To provide a little bit of context, a longstanding goal of the Stanley Center for Psychiatric Research has been to translate genetic findings that underlie risk for severe psychiatric disorders into mechanisms and drug targets. Genetic studies suggest that the overall risk for schizophrenia is largely shaped by variation across many genes, and that it's the combination of genetic nudges in key physiological pathways that likely create a vulnerable system. While cellular models offer the potential for deriving disease-relevant cell types to help elucidate how and in which cell types known risk variants act to produce negative effects of function, we need large sample sizes, and most in vitro models have been limited in the total number of cell lines, which can lead to high variability and confound analysis of genetic variants with small effects. Moreover, conventional experimental approaches have largely focused on investigating effects at a single genetic locus. So we wondered if there was a way to learn from all the genetic influences at once across multiple donor backgrounds. So this led us to the idea of a population experiment in a dish. The crux of this experimental model is to culture hundreds of cell lines from unique individuals together in one dish in a controlled experimental settings. This not only ensures that the cell environments are identical across donors, but it can also be probed and processed simultaneously. Moreover, this setup has the potential for reducing any technical noise that might arise from technical variation that otherwise would have confounded some of the discovery for subtle genetic effects. So how does this experimental model work? In a typical village experiment, we pool together tens and hundreds of genotype cell lines. We then use the natural sequence variation as an intrinsic barcode to identify individual donors within the pool. And we've developed two approaches to map donor genotypes to expression phenotypes. The first is called census sequencing, which is a relatively fast, cheap, and robust technology that relies on low coverage sequencing to quantify the presence of each donor's DNA in the village before and after sorting or selecting for a cellular trait of interest. The second approach for interrogating such genetic effects at scale is called dropulation. And in this case, after pooling individuals' cells into a dish, cells are partitioned into droplets and submitted for single cell sequencing. The donor of origin for each cell is subsequently inferred using transcribed SNPs and prior knowledge of the donor genotypes. Results from such experiments can then be used to look for donor-specific differences in gene expression without the need to control for either batch or well-to-well variation. Now, we describe both of these methods in much greater detail in two preprints that are available on bioRxiv. But in the interest of time, I'll briefly go over some of the key findings from each of these manuscripts. So briefly, in the first study, we were able to successfully quantify and map the effect of common alleles on the SMN protein expression using census sequencing on a village of more than 100 donors. SMN is relevant because it is the protein that underlies the disease phenotype in spinal muscular atrophy. So we did this by creating a pooled culture of 113 distinct iPSC lines. Next, we labeled these cells with a monoclonal antibody for SMN protein and then sorted the cells into high versus low fractions. Now, using census, then we were able to identify the donor in each of these cellular fraction, and moreover, leveraging the donor's change in representation between the two groups as a quantifying phenotype, we were able to detect a very strong relationship between the total SMN copy number and the SMN protein expression phenotype. So here, each dot represents the expression of SMN in an individual donor and its correlation with the underlying SMN copy number, such that the more copies that the donor has, the higher the protein expression phenotype. We wondered next whether census sequencing would be able to pull out this signal agnostically in a genome-wide scan. So when we ran a genome-wide association study of the SMN expression phenotype against the donor genotypes, we actually found that the strongest association signal mapped to the locus on chromosome 5 containing the SMN genes. So in principle, this meant that the genetic basis of this phenotype could have surfaced in an unbiased genome-wide scan without any knowledge of the SMN locus, which truly was a breakthrough moment for us. We were also able to show that cell villages can be used to identify variation in drug responses, opening up the opportunity for pharmacogenomic profiling and patient stratification using census. In contrast to census sequencing, dropulation, on the other hand, relies on single cell data to quantify genetic effects on expression phenotypes. In this study, using a village of similar 100 or more donors, we were able to detect more than 6,000 expression quantitative trait loci in the induced pluripotent stem cell village. This was more than we had expected based on previous studies of iPSCs using arrays and other tissues. And we suspect that this improvement in part could be due to reduced experimental variation in the pool setting. Now, in addition to uncovering EQTLs, dropulation can also help us find cell type specific EQTLs. And shown here are instances of 2 significant hits in the induced pluripotent stem cells. Identification of such cell type specific signals can reveal how candidate risk variants may be contributing to differential regulation of genes across cell subtypes. Finally, we demonstrate how cell villages in combination with genetic perturbation studies can uncover variants linked to disease-relevant cellular traits. For instance, using a Serm iPSC-based village of neuronal progenitors, we observed high levels of inter-individual variation in susceptibility to Zika virus driven by a large effect SNP in the antiviral gene IFIT-M3. Taken together, the 2 complementary approaches I've just described, dropulation and census sequencing, really provide a new scalable experimental paradigm for studying genetic influences on cellular phenotypes across hundreds of donors concomitantly, while minimizing variation in gene expression across lines. Now, an important prerequisite for the success of these village-based experimental models is access to a large collection of genotype cell lines with donor metadata. And here we were really fortunate to enter a fruitful collaboration with CIRM and FCDI, which allowed us to expand our villages of initially 10s and 20 of lines to hundreds. Looking forward, we anticipate that these scalable systems will be part of a new experimental playbook, which will allow researchers to connect genetic risk with specific cellular processes, identify responses to perturbations and drug treatments that may vary across donors, and ultimately identify targetable areas of biological convergence of genetic effects, even in complex polygenic diseases. And with that, I'd like to hand off to my colleague, Ralda Naume, who will tell you more about these resources and some of the challenges in sharing them, as well as the incredible team of people without whom this project would not have been possible. Thank you for your attention and time.
Great, thank you, Dia. I'll share my screen next. Perfect. So thanks again, and sorry we couldn't be there live today. So to recap what Dia just told you about, the main motivation behind establishing the cell village system for us was to develop new experimental systems that could bridge the gap between human genetics and experimental biology by measuring genetic influences in ways that are precise, scalable, and well-controlled, and across different levels of analysis, different phenotypes that include RNA, protein, and cellular responses. We wanted to generate and share enabling resources, both cellular resources and data resources. And Gia has already highlighted the two kind of bioarchive preprints where we have described kind of the two first applications of this approach. So the first step for us to be able to really leverage the cell village system was to have kind of a lot of cell lines to play around with, a large sample size. And so for this reason, we established a human cellular and genetic resource whereby we assembled or either assembled or derived iPSC lines from over 800 unique donors. And these included 450 Serm lines that were acquired through a collaboration with Serm. So we brought in these cell lines in-house, expanded them, QC'd them, cryopreserved them, and importantly, we also generated genetic sequencing data from each and every cell line, which included SNP array data, as well as whole genome sequencing data, 30x whole genome sequencing data, to enable the kinds of applications that Dia highlighted. And you can see the full list of cell lines that we have on our website right here. This is roughly the breakdown of by diagnosis of samples in our collection. You can see that there's roughly a third of the cell lines from control individuals or unaffected individuals without a known diagnosis. Another third is from people with neurodevelopmental or psychiatric disorders, and the rest have a wide range of different diagnoses: heart disease, diabetes, eye disease, liver disease, etc. So far, we have either generated and deposited a lot of data sets. First and foremost is the original, excuse me, the original SNP data set from the 2,169 cell lines that Dia has fully QC'd and she has discussed earlier. This is really useful to be able to query to infer genetic ancestry and kinship. So can give information about relatedness between samples, which is very important for heritability studies, for example. You can also use this data set to infer, to compute polygenic risk score estimates. We've done this for schizophrenia, Alzheimer's disease, and others. And again, all of this data is in the ANVO database already. We've also generated and deposited whole genome sequencing data. 300 data from 300 cell lines are already in ANVO, and we plan to deposit the rest later this year. And this data can be used to look at inherited variation as well as acquired mutation, for example, via reprogramming or cell culture. We've also generated a lot of single-cell RNA sequencing data from over 100 of these cell lines from different cell types, iPSCs, neurons, and NPCs. This is also ongoing. We are generating more and more data that will be deposited as we go. And we've also performed different kinds of phenotype measurements that include growth rates and infectivity. Again, these are available in the publications that we have described. So now we would love nothing more than to enable other labs to also use the village approach. And we are cognizant that it's not very trivial to be able, for any given lab, to be able to generate villages from 100 cell lines. It can be resource extensive. So for this reason, our thought in discussion with Serm was that maybe these villages could be shared, villages of different cell types could be shared, for example, by a Serm hub model that is being discussed in this workshop, which we are very supportive of, and I think would love to help do whatever we can to help make this happen. However, I do want to point out that the legal aspect to being able to share such villages is by any given lab, like even us, is not trivial. So that could be an important point of discussion as well to see how we can make this possible. Finally, last but not least, we'd like to acknowledge a fantastic team who made all of this work possible. It really does take a village, including our partners at Serm and FCDI, including Stephen Lin, Susan DeLaura, Steve Smith, Sean Patel, and Uta Grushammer, and of course, our funding sources, the Stanley Center for Psychiatric Research and NIMH. And I would like to thank you for your attention.
I guess I can't thank Dia and Ralda live, but I hope this was as interesting to you as it was to us to be part of this project that utilized the Serm IPS cell repository. If there are any questions, I could try and answer them, but I probably will have to refer you to the future when we have a chance to check in with Dia and Ralda. But if there are any comments, please raise your hand or use the Q&A. I'll just comment that not only did this collaboration or this work by the Broad lead to an interesting tool, the cell villages, and it also improved the quality of the bank itself with addition, with the genomic whole genome sequencing information that is now available on many of the lines, and also the identification of polygenic risk scores for schizophrenia, for instance, amongst those lines. Okay. We'll proceed to our next talk this morning, which will be by Jackie Mayer, who is a professor at UCSF. Jackie is the chief of the Division of Gastroenterology at San Francisco General Hospital and the director of the UC San Francisco Liver Center. Jackie will be talking about the liver lines in the IPS cell repository. So she's a little unusual in today's meeting, which is really focused on the CNS for our discussion purposes. But I wanted to include Jackie because she is one of the collectors actually of tissues for this repository. And like all the tissue collectors, she was able to receive the induced pluripotent stem cell lines derived from the tissues that she coordinated, and has now been able to use them in her own studies of non-alcoholic fatty liver disease. So please go ahead, Jackie.
Thank you so much. Can you hear me?
Yes.
So thank you, Ute. It's my pleasure to be here. And as Ute has mentioned, the reason that I'm here is to really give you a flavor of my experience, both as a tissue collector and ultimately a user of the Serm iPSC repository, in this case for non-alcoholic fatty liver disease. And since I am not a neurobiologist, I thought that I would give some of my time to trying to explain the disease process that I'm interested in and some of the scientific background, and then follow with our experience with the iPSC line. So next slide, please. So non-alcoholic fatty liver disease is a progressive disease, and it's a metabolic disease that actually is a common accompaniment to individuals who have dysmetabolic states such as obesity or diabetes. And in this group of histology slides, what you can see is that the disease initially begins with the accumulation of fat in liver cells, steatosis, but can progress to a necroinflammatory state, which we call NASH, non-alcoholic steatohepatitis, that can ultimately progress over time to cirrhosis and even hepatocellular cancer. And so the reason that individuals are very interested in studying this disease is that epidemiologic studies indicate that it's highly prevalent. As a matter of fact, it's estimated that 25% of the entire global population suffers from non-alcoholic fatty liver disease. It has, in the US, it's become the second leading indication for liver transplantation. And it's expected to actually become the leading indication for liver transplantation within the next 5 years. So if I could have the next slide, please. So not wanting to make anyone too nervous, if you have obesity and diabetes, your chances of having steatosis is 90% or greater. That said, the progression from steatosis to NASH is slower, fortunately, such that only about a quarter of the people who have steatosis go on to develop more progressive disease. And so it's this, or the risk factors that lead to progression, that has been the source of a number of animal studies as well as human studies to try to determine what the risk factors are that lead to progression and potentially look for therapeutic options. And I just want to make one comment that after many years of animal research, a lot of it can boil down into a figure that I have on the next slide, please. Next slide, please. So this is a representation of liver transcriptomic data from humans, and the humans in blue are both normal individuals and individuals with fatty liver disease, as opposed to mice in red. And the mouse data is actually a compilation of at least 9 animal models, all of which are meant to really examine fatty liver disease. And so you can see that despite the best of our ability, the mouse modeling of non-alcoholic fatty liver disease has been sorely lacking. And so I can't say that this particular piece of information sparked human research in fatty liver disease, but it certainly is fueling the impetus to look more at direct human models of this disease. So next slide, please. So as far as the human epidemiology is concerned, Nafld is known to be a heritable disease, and the first GWAS study of Nafld was actually performed in 2008, and it identified PNPLA3 that you can see here in the top right of the Manhattan plot as a risk allele that is highly prevalent in individuals who have this disease. Now, scores of GWAS studies have been performed since then, and most of them are confirmatory of PNPLA3, but also have come to uncover at least 6 and as many as 12 or more additional variants that are associated either with disease progression or disease protection. Now, the reason that I show this particular GWAS study is that it was actually based on individuals who have biopsy-confirmed disease at the beginning of the study. And what you can see here is that there are at least 4 genes that are identified as having a major association with fatty liver disease. 3 of the ones that are highlighted in blue in the Manhattan plot are associated with disease progression, and 1 is associated with disease protection. So I want to also mention that most of the GWAS studies, despite their numerical power, have largely focused on European populations. And that landscape changed just last year, if I could have the next slide, when a GWAS study of the Million Veteran Program in the United States was published in medRxiv. And so the investigators who worked on this study looked at 90,000 individuals in the Million Veteran Program. And as you can see here, they have uncovered 77 risk loci, as opposed to the usual 12 that have come up from the European cohorts. And so this actually is completely changing the landscape and actually encouraging people to think much more broadly about the genetic risks that might be attributable to this disease if one looks at populations that have more broad ancestries. So if I could have the next slide, please. And I just wanted to make one more point before I go on to my personal experience, and that is that as much as we know now about the genetic risk factors for non-alcoholic fatty liver disease, there is still an environmental factor that seems quite important when looking at human individuals in epidemiologic studies. And so what you can see in the graph on the left is the percentage of liver fat, which is on the y-axis, that's plotted against body mass index on the x-axis. And it's stratified by individuals who have different alleles for, in this case, PNPLA3. And so the MM variant are the individuals who are homozygous for the risk allele. And what's clear from this graph is that even if you are homozygous for the risk allele, you don't begin to manifest disease unless you're also overweight. And so the similar data have actually been identified in additional studies, and I just have an example of that on the right. So this is a study that was very similar in its design, but actually looked at a 12 gene risk score, stratified it by BMI, and also showed that even if you are at the top 1% of a genetic risk profile, that doesn't really manifest clinically unless you are also obese. And so this combination of genetic risk and environmental risk is something that has very much intrigued us. And we were interested in determining whether we could apply these two principles to a cell-based model of non-alcoholic fatty liver disease. So if I could have the next slide, please. And why don't you go to the next one? So as Ute mentioned, we were fortunate enough to be funded by CIRM as the tissue collectors for the liver disease group. So this was a program that actually launched in 2013, and we began recruiting patients in 2014 and completed recruitment at the end of middle actually of 2016. So hindsight is 2020. When we began this study, hepatitis C was the most common liver disease in the world, but with the advent of antiviral therapies, it has plummeted in its importance in global health. And so non-alcoholic fatty liver disease has actually become much more important. Still, when we were in the midst of recruitment, we managed to recruit more than 150 liver disease patients, as you can see here, about 2 to 1 of hepatitis C and Nafld. And at the same time, we were asked to recruit normal controls. We were restricted, however, in the number of controls that we were allowed to recruit, and were instructed that the controls that we recruited needed to be suitable to serve as control for not only our cohort, but other disease cohorts that were in the California network. And so ultimately, we managed to collect either PBMCs or skin biopsies from 184 subjects. And about 80% of those cell lines that were generated by CDI are now available at Fuji. So if I could have the next slide, please. So as Ute said, we were fortunate enough to be able to get these cell lines back. And so we have focused specifically on the Nafld lines. And so I want to give you a little bit of the demographics of the cohort that we're working with. So right now we have in our possession 44 Nafld lines. All of these subjects that were submitted to the IPSC repository had biopsy-proven diagnoses with highly annotated clinical data. I happen to work in a hospital that is in a Latinx neighborhood, and so our population is enriched in Hispanics, and they have a high frequency, as you might expect, of PNPLA3 risk alleles, with almost all of them having one allele and over half of them being homozygous for the risk allele for PNPLA3. As you can see on the graph on the right, though, if you look at the number of risk alleles for other genes, and so here I've shown the 4 sort of usual suspects in the Nafld space. What we find is that a good number of our subjects have multiple risk alleles, with the average being about 3.9. And so PNPLA3 is clearly not the only risk allele that is present in this population. And furthermore, if you look at our control subjects, which actually come from a variety of ethnicities, even they have an average of 2 risk alleles for these 4 Nafld specific risk genes. So if you could go to the next slide, please. So we decided that we wanted to go bold and have as much of a discovery experiment as we could as a first stab. We were unable to actually look at the entire cohort because of resources and manpower. But we decided that we would do as many, or sort of differentiate as many of the cell lines that we had as we could to hepatocytes, primary liver, or hepatocytes liver cells, and then look to see if we could determine whether there are any differences either in transcriptomics or in phenotype between the lines that came from patients with disease as opposed to those who came from healthy controls. And so I just show you here a snapshot of what the differentiation protocol looks like. It's about a 22-day event. in which the cells go through a state of definitive endoderm and then finally get to iPSC-derived hepatocytes. And so what we did initially was performed RNA sequencing on the parent iPSCs and also on the iPSCs at the end of differentiation. So next slide, please. So we were actually very surprised, but thrilled to find that when we differentiated these cells to iPSC-derived hepatocytes, we could immediately see that there was a visual difference between the cells. The cells from Nafld patients looked as though they had vacuoles under standard microscopy. And when we performed Vodipy staining, it was clear that this was evidence of increased lipid accumulation in green. And so actually further studies went on to show that biochemically they have excess triglyceride. And I want to emphasize that these cells were not treated with anything in order to challenge them metabolically. This was just under standard culture conditions. If you can go further, please. One slide. And so when we looked at the transcriptomics of these cell lines, we found that they segregated into what I think are three different cohorts. Clearly the healthy control cell lines on the right are different from the cluster in the center, but the cluster on the left looks like it has somewhat of an intermediate phenotype. And so I just draw your attention to the SNP genotyping that we performed at the bottom. So the cluster in the center, which is a disease, the primarily diseased patients, certainly has an enrichment in individuals who are PNPLA3 homozygotes. However, it's really not 100%. And as you can see, there's a smattering of PNPLA3 homozygotes in both the normal controls as well as the individuals who are in the intermediate cluster. And so what did these data tell us? I think there's a couple of things. First, the fact that we got a transcriptomic and a phenotypic difference when differentiating these cells without doing anything else but putting them through a standard differentiation protocol actually really much energized us to the concept that whatever is going on genetically in these cells is actually separable from the environment. And so we've taken the environmental influence out of this and feel as though that there is a distinct genetic background that is driving this difference in phenotype. And furthermore, because of the graph that you can see at the bottom, clearly the usual suspects in terms of genetic risk factors may not be the entire explanation. And so there may specifically be genetic risks that we don't know about yet that could be underpinning the differences in these cell lines. And so just if you can go to the next slide and actually go ahead to the final slide. So what are the opportunities and challenges that this experience has taught us? I think, as I mentioned, our experience to date, albeit we have very preliminary data, but it's exciting data in our view that really spurs us to want to do more. One thing that we could do, and is already being done by other laboratories, is to generate multicellular Nafld co-cultures that have not only hepatocytes, but other liver cell types to see if we can look at downstream factors such as inflammation and fibrosis. Another thing that's clearly on our minds is to perform CRISPR gene editing to either correct the risk alleles that we know exist, either singly or in combination, and then try to deconvolute the results that we have to see which genes are responsible for which phenotype. Third, we really feel as though when we correct some of these lines, we're still going to get some that behave abnormally, even if we think that they're genetically normal. And so that's going to spur us on to do some more discovery experiments to see if we can find new genes that might be related to disease phenotype. And finally, to expand the cohort so that we can look at individuals who have the same clinical disease, but belong to different backgrounds. And so finally, what are the challenges? I think we have been posed with two major challenges in completing this work. First is that the cohort is underpowered, which is unfortunate. We really do need more cell lines in order to be able to convince the scientific community that what we have observed is a real event. Second, I think it was a lost opportunity to not have controls that are matched for our patient population in both, ethnicity, gender, weight, even genotype. And so that's something that I think needs to be done. And certainly there are other things that we can do as far as, the level of disease, so the severity of disease. There are many different factors that we can include in a larger cohort, but all of this speaks to the need for increasing the number of cell lines that we have to work with. And then finally, as has been mentioned, this work has been expensive. It's been challenging for us to accomplish as an individual laboratory, and it's been challenging to convince people to get behind in terms of support. But we're hoping that we can, continue to push forward and get some things done with these cells in the future. So thank you very much.
Thank you, Jackie. As somebody who was intimately involved in getting this IPSL initiative initially launched at CIRM, it's very exciting to see where this has gotten us in terms of you being able to start to look at at this collection. And I hope that ultimately you'll be able to use it to really pinpoint the contributions of these various risk variants to liver disease. Which would be a fabulous outcome, of this collection. We have one question that came in and from Mike Snyder. What is the proportion of the heritability for Nafld, is I think how you call it? What proportion has been mapped by GWAS?
I do not have my hands on that information, but I can certainly try to get it for you in the next and have it, for the next discussion.
Okay. Wonderful. And another question that came in, you are probably calculating polygenic risk scores for your donors. How does it compare with the underlying diagnosis in the ability to stratify the cohort?
So, again, the numbers are so small. Within each of our clusters, I think it's really difficult to draw conclusions about the impact of a polygenic risk score in a cohort that really is only 10 individuals. And so as much as I would love to say that this is predictive, we haven't been able to make a determination that is associated as yet. It certainly doesn't look from our transcriptomics to date that the individuals who have the highest genetic risk score are the ones that have the worst disease.
And then sort of a related question about the pathways that are implicated by these risk variants. Is there any commonality suggested, by the various risk alleles to explain this phenotype?
Well, clearly they're all related in some way to metabolism, but there are some specific differences. So PNPLA3, for example, is a lipid droplet protein that probably has something to do with lipid hydrolysis from lipid droplets. TM6SF2 has a lot to do with lipid export from the liver because it's part of the VLDL secretion program. GCKR has everything to do with glucose metabolism. So there are individual differences for each gene.
I see Clive has his hand raised.
Hi, Jackie. Great talk. Fantastic to get a phenotype like that just spontaneously in the liver cells. You mentioned problems with the controls. Seems like there are 300 controls in the Serm repository. Are you limited in getting them from Serm, or why are you limited for controls? I just didn't understand that.
Well, I think that where our critics have had a problem is that they want better annotated data, but you're exactly right. There are a number of healthy controls in the cohort. And so I think we probably just need to mine the cohort more fully to see if we can match individuals better to our group. And so, actually today's information is going to be helpful in that regard.
Did you match male-female pretty carefully? Because we found a lot of male-female differences coming out now in motor neuron differentiation, which we were surprised by in terms of genomic changes.
Right, and the answer is we did not. So our control cohort is 50-50, and our Nafld cohort is about 40-60, but it was not intentionally matched.
Thank you.
Okay, thank you, Jackie. We'll hear more from you later in the discussion, and we'll move on to the next talk now. I'd like to introduce Kaikas, who is Chief Exploration Officer and Head of Neuroscience at In Sitro. And he'll talk to us about his organizations and his work on machine learning and engineered iPS cells for unraveling the complex biology of CNS disease. Please take it away.
Thank you, Uta. It's great to be here. It's great to see familiar faces. For some of you, I think for Clive, this talk will look pretty familiar since I gave a similar talk at the California ALS Summit recently. Can everybody hear me?
Yes.
Okay, great. Next slide, please. So In Sitro, really a lot in kind of the name says it all. We're really a company that focuses on a fusion of in silico and in vitro, very different environment where we're really focusing on using data science and machine learning to really understand complex biology, and have been at it for about 3.5 years, really focusing on building a company that has an integrated culture of data science and biology. Next slide, please. Skip to the next slide, please. I have a few extra slides, so I'll try to go faster with some of them. So really at the center of what we're trying to do at In Sitro is build a bridge between in vivo and in vitro data using genetics. And so a lot of the resources that were described already from Serm really help us do that, particularly on the in vitro side, where we can take iPS cells and differentiate them to different cell types, and then connect up through genetics to in vivo phenotypes. We've been more and more focusing on high content data, really instead of just single biomarker data, going for high content imaging data or large scale transcriptomic data. And really the goal here is to build a disease state manifold for any given disease where we can use that to generate predictive models and then do screens. And I'll go through some of the examples of that, but we have programs in liver and in neuroscience. Next slide, please. And so we've, over the 3 years, have really built up 3 areas of focus, one on translational machine learning, using statistical genetics and machine learning to help us identify targets and stratify patients. I'm going to go through some of the work on predictive disease models, where we use iPS cells to generate disease models, to look at cell state, to help us prioritize targets. And recently, we've started to work on molecular design using machine learning for instance, DNA-encoded library screening. Next slide, please. I'll skip to the next slide. So one of the first projects that we've launched into is a monogenic disease, and we really did this to kind of carefully build our platform. And for iPS disease modeling, as you know, that identifying phenotypes can be quite challenging for many diseases. And so starting with something that was a known pathway, the Mtor pathway, where there was clear genetic drivers was key. So we started with a disease called tuberous sclerosis complex, which is really due to mutations in TSC1 or TSC2. This leads to benign tumors, and really the major morbidity is in the brain, where individuals have epilepsy, and kind of surgery is the common way to treat individuals. There are, for us, it's actually very important that there are drugs that are used to revert the phenotypes, rapalogs, everolimus. This is effective in about 50% of the patients, but there's still a lot of opportunity to further pull apart the pathway and really generate therapeutics that help the additional 50% of the patient population. Next slide, please. So there are known biomarkers that are used for the Mtor pathway, shown on the left. So if you knock out TSC1 or TSC2, you can actually measure activity of the pathway by measuring phospho-S6, which is one of the downstream pathways for Mtor. And what you're seeing is just an iPS line below that was turned into cortical neurons, where we either have wild type or knocked out TSC2. This was done on control Serm lines, where we've engineered this in with CRISPR. Very importantly for us, we've started to go a little bit deeper. And this is really because for many diseases, we don't know what the pathway is. We don't know what the phenotypes we're looking at. So we've started to build up the capability of to look at morphology, to look at deep transcriptomics at the single cell level, along with doing longitudinal live cell imaging with quantitative phase microscopy, and actually be able to look at kind of deep phenotyping over time. Next slide, please. So this is just one example of experiments we've done with the TSC2 knockouts, where we've taken either TSC2 or wild type, which you're seeing on the left, and done single cell RNA-seq on this. This single cell RNA-seq is pretty common these days. And what you can clearly see when you take all of the genes and do a UMAP and project into 2 dimensions, the TSC2 cells that are a knockout cluster very separately. If you treat with rapamycin, it brings them closer to the wild type cells, which are in orange and red. And a few of the genes that we've been studying in the pathway, if you knock them out, you can actually revert the phenotype, which are good places for us to go in terms of drug discovery. We've also been able to generate some machine learning models on the data, do a LDA projection where we actually can generate a disease axis from wild type to disease, and then use that data to then project onto in vivo samples. So we were able to get some in vivo RNA-seq data from individuals that actually have tuberous sclerosis. And what this data is you can get healthy tissue from around the tuber in an individual's brain, and actually look at whether the predictive model that we've generated in vitro can actually predict whether the samples are wild type or disease, really allowing us to connect up the in vivo in vitro data. But we want to actually do this a lot more as we move forward with additional types of data, imaging data and live cell data. Next slide, please. We've also just been able to look at, when you treat with rapamycin or when you treat with some of these gene knockouts that we're now studying, whether that pulls the disease cells back closer to healthy on our disease axis. And what you're seeing is that rapamycin turns the disease cells, as you'd expect, closer to healthy. And some of these additional mutations that we're looking at, these additional genes actually do the same thing. So just really, kind of confirming that we can study this pathway using RNA-seq. Next slide, please. We wanted to further just validate that the RNA-seq data was also reverting the phenotype that we see with phospho-S6. RNA-seq is a very useful tool, high dimensional data, but also just wanted to confirm that you can actually revert the phenotype when you actually look at the known marker. And that's just shown here with the wild type cells. And then TSC2, you can see phospho-S6 going up there. And then when you actually treat with rapamycin, it goes down, or when you knock out this additional gene that we're working on, you see the phospho-S6 go down, really just confirming the same results from RNA-seq. Next slide, please. Very importantly for us, we wanted to step away from transcriptomics and also marker-based data to determine if we have a disease where we don't know what the pathway is or what the phenotype is, can we actually take a high-dimensional data, something that's very cheap and fast, and use that to classify the cells. And so we generated images of all of the neurons, either wild type or mutant, or treated with rapamycin or additional gene knockouts, and fed this into a convolutional neural net, and then trained a machine learning classifier to classify either sick or wild type just based on morphology. And we're able to very well actually classify whether the cells were sick or healthy on the right based on the machine learning algorithm. And this gives us really a powerful tool to then go beyond TSC and start to look at other diseases using just morphology alone. Next slide, please. In addition, we've been building up the capabilities to do live cell imaging so that we can do longitudinal imaging, and we've built a platform to do quantitative phase imaging. This is a digital microscopy that actually allows you to do some very creative things where you actually take the images and you hallucinate, for instance, a particular marker in live cells. And that's what you're kind of seeing at the top with the cell tracking. We can actually hallucinate the nucleus or ER or other features. And you can actually take labeled images and then actually train a computer vision algorithm and machine learning algorithm to actually then hallucinate whether that marker is there, which gives us a very powerful tool to kind of go deep on live cell imaging. Next slide, please. So we used this to see if we could classify the wild type and the mutant TSC2 neurons, either treated with rapamycin or the additional genes that we've been studying. And what you can see here is over time, at the top is the TSC2 knockout with a very clear phenotype. If you treat with rapamycin, over a few days, those turn closer to wild type. And then additional genes that we're looking at here revert the phenotype also. So this gives a very useful tool, and we're really expanding on this to start to be able to clearly look at different structures within the cell in live cells so that we can actually really go deep on biology and other diseases. Next slide, please. So in addition to tuberous sclerosis, we've been focusing on ALS. We have a partnership with BMS. And here we're really interested to see if we can use some of these unbiased methodologies to really look at ALS very deeply. And from the genetics of ALS, it's very clear that at least for the familial form of ALS, there are multiple genes that are clustered into pathways. And we'd really like to be able to see, can we actually, in an unbiased way, cluster cells from individuals that had ALS into these different distinct categories of different biologies. And the goal here is really to build a manifold of ALS, starting with the familial disease, and then really try to understand sporadic disease, on top of that. Is it really through the same mechanisms? We know everything converges at TDP43, or most of disease converges at TDP43, but are there really different biologies here that we can get by going deep here? And so for this project, we've taken a lot of lines that we've actually got from CIRM, control lines, and we've engineered on top of these control lines all of the known genetic drivers for ALS, about 30 different mutations. And we've done this, some of them we've done in just one line, but a lot of them we've done actually in six different CIRM lines. So really creating a very large bank of isogenic lines that we're going to use to generate this phenotypic manifold, along with getting sporadic lines and control lines, in addition from AnswerLS, from Clive's and Clive's group. So really hoping to go deep here and really kind of uncover commonalities between different forms of ALS. Next slide, please. These are just the different mutations that we've engineered, and starting to differentiate motor neurons for these. Next slide, please. So to accomplish this, we've also invested heavily in making things robust and automating. We have a couple different work systems. This is actually work that I started many years ago at Novartis and is kind of extended to in vitro, building up the ability to automate IPS culture and differentiation. And one of the challenges when I was at Novartis is just data capture, and the programming that one needs to do to actually get this to work is really challenging. But in vitro, it's gone a little bit faster and better because we've got a really integrated team that does software engineering and we're able to track data. But so far, we've been able to really onboard 24 IPS lines at once and basically passage them, characterize them, taking images, and then differentiate them to motor neurons. You can see that on the next slide. So this is one of the maiden voyages that we've done, where we've taken just 6 control lines across 4 replicates and then turn these into motor neurons using a directed differentiation protocol. And what you see on the right is single cell RNA-seq RNA velocity over time, looking at the different populations of cells that we generate, along with looking at reproducibility across those lines. We're getting about 50 to 60% motor neurons, and this seems to be quite reproducible from line to line, from batch to batch. Next slide, please. The colors have gotten off when you go from Google Slides to PowerPoint, but it's not super important. So really the goal here is to develop predictive cellular models for ALS, to really be able to train and test machine learning on the familial forms of ALS, where we've got really strong genetic drivers. I compare that to control motor neurons and really determine whether we can distinguish familial from ALS, and whether there are different clusters of familial forms of ALS like people think there are. And then to use that to generate a manifold to then plot on top of that the sporadic disease, to see if we can learn about sporadic disease by, through the lens of familial ALS. And hopefully through this process, we're going to be able to generate very robust models that then we can go on to target identification and test some of the known genetic drivers, additional genetic drivers of ALS, along with doing chemical screens. So really trying to go deep and make a very robust model for ALS. Thank you. I think this is it.
Thank you, Aj. Again, very interesting use of, if I may focus on the Serm lines as one thing, I'm trying to pay attention to. And of course, you've taken these control lines for these motor neuron projects, to an incredible level, of analysis. One comment I wanted to make is with regard to the use of the Serm IPS cell repository. Ralda and Dia's talk and your talk also sort of illustrate how much interest there has been in just obtaining the control lines, but there's, they have diversity in them. There's a lot of them, and then to engineer on top of that, and that's of course a very powerful way of using this repository and doing these kinds of studies. And so we'll talk more about that in the discussion today. I'll also just say that the things we've heard so far, have raised a lot of the issues and points, we're going to discuss further this afternoon. I have a question that came in. Somebody's curious whether you measure phenotypes over time as well when you look at these cultures.
Yes, we are really starting to go deep there and actually look at phenotypes over time with the quantitative phase imaging. That's something that one can do. You're a little bit limited to the types of phenotypes you can measure, but that is something that we're really heavily investing in. And what you need to do that, if you want to look at specific phenotypes, is actually have a reporter line that has a particular marker. You can then train a machine learning algorithm to recognize that, and then you can actually hallucinate that in a non-labeled cell. And we've actually been doing that quite a bit. We have a bunch of new data there. So yeah, definitely very much interested in being able to do longitudinal live cell imaging on phenotypes over time.
Mm-hmm. Okay, thank you. And we'll get back to some of the things you said later in the discussion. We're doing well on time, I think to a large extent because Rosa and I were so well-timed this morning. But we'll now go and have the last talk of this morning's session, and we'll switch to a use case of the Serm Genomics Stem Cell Hub. And we'll have Aparna Baduri, who is now an assistant professor at UCLA, speak about the characterization of cortical organoids she led when she was a postdoc in Arnold Kriegstein's lab. And the goal is manifold of having this talk now. Obviously, it'll be a great lead-in to this afternoon's discussion about characterizing organoids with great detail. But I also would like, or the purpose of bringing in Max Häusler as well. Max is an associate research scientist at UC Santa Cruz. Purpose is also to highlight how an experimental computational collaboration came, out of the Genomics initiative as an example, perhaps, of what we would also like to foster in future research initiatives at CIRM. So Aparna, if you want to get started, please.
Thank you, Uta, for the invitation. And I'm really excited to talk with Max today about some of the work that we have been doing in the Kriegstein lab with a great set of collaborators, including Arnold Kriegstein and Tom Nowakowski and Alex Paulin, who are the initiators of this CIRM grant, and how the data that we've collected from this repository has really been broadly distributed and made available both for our own internal use as well as for the community through these collaborations with Max Häusler at UCSC. Next slide, please. So very briefly, as you know, the human brain is a very complex structure. And the outermost layer, the cerebral cortex, is divided into functional areas, some of which are represented here. And each of these areas has unique distributions of cell types across the 6-layered structure, and also has unique vulnerabilities to various neuropsychiatric, neurodevelopmental diseases, as well as other responses to injury, etc. Now, understanding how these areas and how the cortex in general emerges is one of the big questions of human neurodevelopmental biology. And we seek to understand this by looking at both primary tissue as well as modeling this process in cortical organoids. Next slide, please. Here's just a very brief overview of cortical development. You see that there are radial glia progenitors, which are the long cells that go through the entire span of the cortex initially, and they give rise to subtypes of radial glia, which are important for some of the expansion of the cortex and the diversification of cell types. These radial glia give rise to intermediate progenitor cells, represented as the green circles, which then generate newborn neurons, which will become important later in the talk. These newborn neurons migrate to the cortical plate, where they birth the deep layer neurons and then the upper layer neurons. And once neurogenesis is almost complete, the radial glia progenitors go on to engage in gliogenesis, generating many of the glial populations of the cortex. Next slide, please. So today I'm going to overview a set of papers that were part of this large collaborative effort in the Kriegstein lab. So this one was led by Tom Nowakowski, and this was the first profiling effort using single cell sequencing that broadly sought to identify the census of cell types that exist in the developing human brain. And I won't go through all of the experimental details because each of these studies has now been published, but I will highlight some of the aspects of this data that have been really powerfully shared with the community and that enabled aspects of this collaboration via the collaboration with Max. So this first study, we sought to profile different cortical areas of the developing human brain and generated a data set of about 4,200 cells. And as you can see from this plot on the right, we were able to identify a number of subclusters as well as a number of major cell types. Next slide, please. This was the first data set that we were able to engage with Max on to start to develop the UCSC browser, and you'll talk about some of that process later on in the talk. But the main finding from this study was that when we were looking at the metadata, such as the area from which the cells were known to be dissected or sampled, we saw a segregation based on this sampling area in the maturing neurons only. And this is shown towards the bottom of the plot in the purple and the blue. And this suggested to us that there might be early specification events that are determining PFC versus visual cortex identity. Next slide, please. And this led to a model where we initially described this process of cascading identity, where a small number of differences in the prefrontal cortex are expanded through differentiation and become more pronounced in the maturing neurons. Now, this was a baseline study and was the start of some of our comparisons to cortical organoids. So next slide, please. Next slide, please. And this began a collaboration between myself and Madeline Andrews, where we sought to profile both the process of cortical development in cortical organoids and compare that to some of these atlas scale data sets that we were generating, both part of that paper as well as part of ongoing efforts in the lab funded by the Brain Initiative to create an atlas of mid-gestational human brain development. And really what we were looking to see if we could make comparisons between aspects of cell type specification, maturation, and areal identity. Again, because this is published, I won't go through all of the details, but next slide, please. We did profile 5 individuals and about 200,000 cells from 7 cortical areas, ranging from gestational week 6 to 22. And this was our primary reference data set. And as you can see from these single cell plots here, we were able to identify many of the major cell populations we expected, including SOX2 progenitors, HOPX outer radioglia, a subtype of radioglia expanded in the human brain, EOMS positive intermediate progenitors, NeuroD6 and SATB2 positive neurons, and DLX6 AS1 positive inhibitory neurons. We compared this data to, next slide, please, organoid cells that were obtained from 4 different stem cell lines, including iPSC and HESC lines, using 3 different cortical organoid generation protocols. We surveyed a time period from 3 to 24 weeks, which should roughly encompass the same time period from primary development, and profiled about 240,000 cells. As you can see, we found the same primary major cell types, although there were some discrepancies in their overall proportion that was represented. Next slide, please. Using this data, we performed a number of comparisons between the organoid and primary clusters. And I won't go through all of the details, but as you can see, some of the clusters, especially the dividing clusters, were highly preserved between systems, while in some cases we saw more of a pan-radioglia or pan-neuron identity in the organoid compared to the cleaner subtypes that we found in the primary tissue. Next slide, please. This is quantified here by the number of high-quality subtypes that were identified by each broader cell type, and by the quantification, where we saw a very strong preservation of state and class, i.e. dividing or non-dividing, neuronal, non-neuronal. But in terms of some of the more specific types and subtypes, we saw a decrease in the subtype specification that was observed in the cortical organoids. Next slide, please. In an earlier comparison in a project headed by Alex Paulin and also part of the Kriegstein lab effort, we had previously looked both at organoids from human as well as from other non-human primates. And through this study, wanted to make a comparison between the primary tissue and the organoid tissue. And one of our primary observations was an upregulation of glycolysis modules in the organoid compared to primary tissue. And so in this follow-up study, Madeline and I tried to validate some of these observations. Next slide, please. And what we observed, I'm sorry, I know this is a busy slide, is that we were able to immunostain for markers of glycolytic and ER stress, including PGK1, ARKN1, GORSB2, also across data sets, these genes were higher in some of the organoid systems from different published studies, including our own, to compared to primary tissue. Next slide, please. So as part of this study, we performed a number of experiments to explore where the source of the stress was and whether it was related at all to cell types, cell subtype specification. And one of the experiments that I will highlight is how we dissociated weak 8 organoids and transplanted them into the mouse cortex. And after several weeks, 2 or 5 weeks, we were able to use FACS sorting to isolate them from the mouse brain and perform profiling on them. And below is shown an example of how the human cells integrated into the mouse cortex, or rather sat on top of the mouse cells. Next slide, please. When we did these transplantations, we did see very complex morphology, which is something that's very interesting and we are now following up on. But we also saw that there was a significant downregulation of these stress markers, both at 2 weeks post-transplantation and 5 weeks post-transplantation. Next slide, please. This was quantifiably significant, both based on the immunostaining and the single cell sequencing. Next slide. And resulted in an increase in the cell subtype specification. Next slide, please. So this comparison between cortical organoids and primary tissue offered us an opportunity to really leverage some of the data sets that we had been using. And again, all of those data sets that we have looked at have been highlighted on the browser devised by Max Hausler. And so I'd just like to highlight some of the more recent efforts that have expanded on this data set. And so this is a more recent atlas of the developing human brain, and especially looking from the first trimester through the 3rd trimester. So I'll just give you a very brief sampling of this data on the next slide, please. where we again were able to profile more areas of the cortex where we wanted to, and regions of the brain, where we wanted to more precisely identify the timing and the cell type specification programs that are specific to each of these cortical areas and brain structures. So next slide, please. This study expanded the number of cells that we had across cortical areas to closer to 400,000 now. And as you can see, we were still able to identify the main populations that we had previously seen, both in the 2017 study, as well as in the comparison between organoids and primary tissue. And importantly, this data set has also very recently been added to the UCSC cell browser. Next slide, please. And with that, I'd like to hand it off to Max Hausler, who will talk more about the aspects of the collaboration and how the browser evolution process happened and has been used by other groups to leverage these and other data sets within that environment.
Yeah, thanks, Aparna, for this impressive presentation. And thanks for the invitation here for being able to present the trivial, easier software part of it. I'm not an expert in neuroscience or single cell, but I work on the UCSC genome browser, and you may have used that or another tool I wrote, Crispr, which is a Crispr designer. So I've spent 15 years on developing user interfaces as a software developer. The data browser here, next slide, please, was for the single cell data. This talk would actually mostly fit into tomorrow, because this is now about the data infrastructure more than about cell line infrastructures. But it fits also very well into here because we applied it was for this particular use case. So CIRM is trying, for the data infrastructure, is trying to make data accessible, the FAIR principles, findable, accessible, interoperable, reusable. And in many ways, the existing CIRM data warehouse, CIRM.ucs.edu, does that for the CIRM Genomics initiative. You can go there. There's a typical faceting thing on the left, just like on Amazon. You check the boxes you want, the types, and then you can search for files and download the files quickly. So that is findable and accessible. But for the single cell data, is it sufficiently accessible for people, what people are expecting? Next slide, please. Because what most people want are these plots from the paper, right? The paper is characterized 20,000 genes, but you would ideally, you don't want to download gigabytes of data in a big data environment or supercomputer. You want just a couple plots. And so I had a quick go on just a quick hack. This is still on the CIRM data warehouse today. It's embarrassing, I'm not pointing out the URL. But the Kriegstein lab really liked that for the single cell data, and it was an initial first shot, and then I spent 1 or 2 years adding more features to that, guided by Aparna, Tom, and the other members of the Kriegstein lab. Next slide, please. And the result is this tool that has 1 page per data set, and the first data set was the initial paper that Aparna just presented about developing human cortex single cell data. And the users, the initial 2 or 3 users, and then more people over time contributed all these ideas and all these buttons on the screen, and the different features and how they interact. So I didn't know anything about single cell at first, but then of course we all together learned, and I think it was a very efficient way of designing this user interface based on actual users. Next slide, please. And the features were very much guided by this particular CIRM project. So there was limited funding for this user interface. We knew that the funding would go down after a while. So it had to be cheap to run. And we knew we had several data sets coming in, so it had to be generic enough. And because most of the programmers, well, the 1 or 2 programmers that I don't know enough about single cell analysis, and Aparna had all their analysis already done. So it was very clear that I would only import results. I would not re-analyze, which made it a lot easier. And the data sets stand on their own. They have their own addresses, internet address. So you can find them in Google Scholar, and I will talk about that more. And there's a lot of documentation now on setting this up your own, because it's so small, you can run it on your own web server. There's no backend server. That was for cost reasons. Next slide, please. And more and more, there were more features added. It's by far not complete, but it's always nice for user interfaces to add more things, but there's a couple split screen mode and things. Heat map viewer is a standard one. there is still much more to do. Next slide, please. But we focused quite a bit of time on adding more data sets, because people, after these, you saw Nature Science. These papers have been cited 800 or 900 times now. Other referees, journals, and authors asked us to add their data sets to the Cell browser. And while today we have 100 data sets, mostly using one FTE, Matt, a colleague of mine, a large part of the usage is still Kriegstein CIRM data sets. But there are many others now, and it happened to become one of the primary biggest collections of single cell data sets in a consistent format. Next slide, please. So it's relatively popular. We have 3,000 monthly users, and the single cell browser has been mentioned in 180 papers. And I looked for this presentation a little bit on how people cite what does a typical paper look like that's mentioned a single cell browser URL. Next slide, please. And very often, these are papers that use the website. It's not just a drive-by citation that Lior Pachter would call it. It's a paper that actually used the data for something. And usually they say, we looked at this and that gene on that website and we concluded something. So that is Aparna's original paper has now 600 citations, I think, and around 64 of them cite the URL. So that gives us an idea of how people use the data. Next slide, please. So for CIRM, because one of the big questions for this panel and for the panel discussion, for at least for tomorrow for sure, is what should CIRM do about data? And I think this example illustrates quite well that yes, there is a case for big data analytics and supercomputers and cloud computing, but many users are not in that space. Many researchers want to go somewhere and click somewhere. And a visualization portal, something that shows something with very little cost, with a very small barrier of entry, I think could be useful. What helped us here was that CIRM mandated the data sharing right from the beginning. So for the CIRM Genomics Initiative, a very particular rule, the data warehouse got the data first, then the labs got it. But I think the Kriegstein lab would have shared it anyways, of course, early on that worked pretty well. It helped that the Kriegstein, Aparna and colleagues were extremely successful, as you saw, and knowledgeable, and were able to drive this very well. And that as the Jim Kent's group, I and colleagues, people have been doing visualization for 20 years, are staff scientists, and so there was definitely no competition about papers or anything. And the staff scientists like me, the programmers, are of course much more expensive than postdocs, but were covered here by CIRM, and Stephen Lin and CIRM encouraged this. So the second version, the final cell browser, was really encouraged by CIRM all the time, and that definitely helped. And so that's pretty much what I wanted to say. It was driven by the users, it was driven by CIRM, by the world's experts on single cell. And I hope that for other data sources we've seen in the other talks, microscopy images, variants, for all of these, there could also be a superficial, easy interface where you can explore what CIRM data has to offer and then decide on what you want to download, what you want to run on. Thanks.
Thank you, Max, and thank you, Aparna, for illustrating one small example that came out of the Genomics Initiative. It was a much bigger effort, of course, but I really like when I learned from you all how this collaboration between experimentalists and software engineers like yourself came about. So thank you for illustrating that. We had Matt Blurton Jones, you raised your hand earlier.
Yeah, sorry, it was an accident. No, sorry, it was an accident.
Gotcha. And then we have a question as to whether the browser, the cell browser tool, can be used for all single cell data sets, for instance, ENCODE.
We have some, yes, we have the mouse ENCODE data for sure. We have not added all data sets just because there is no comprehensive collection of single cell data sets yet. and NIH hasn't made one. So, yeah, but yes, there's 100 data sets on the website, and one of them is ENCODE mouse.
Thank you. All right, it's like 1 o'clock. It's amazing. In terms of punctuality, I will recap a little bit of what happened, what we heard this morning when we start our discussion this afternoon, which I look very much forward to, starting to look into both the scientific challenges that lie ahead in terms of continuing shared resources, and also into the practicalities of how to build the next generation of shared resources labs. So I'll end right here, and will ask that all the panelists and attendees, we do have now a break until 1:30 Pacific time. I suggest everybody come back a few minutes early so that we can start our discussion at 1:30 promptly. Thank you. Okay, it's 1:31, so let's get started. Welcome back from the break. This morning, we heard an overview of previous CIRM-funded research resources with specific examples of how these resources have been used. So this afternoon, we are going to look to the future and gather input from a most stellar group of experts that will inform CIRM about potential opportunities to share resources and promote collaborative research through the creation of competency hubs. Next slide. I just spoke to the slide highlighting the fact that the focus of this afternoon's discussion will be these competency hubs. Next slide. So you might ask yourself, what are competency hubs? So we envision a competency hub to be an entity that shares a specialized skill or resource at any stage of the drug development pipeline with other investigators in a collaborative manner. So when I say we, that's CIRM. And as we envision going forward and creating a multitude potentially of competency hubs. Next slide, please. Today's discussion, though, will be focused on one specific kind of competency, and that is stem cell-based cell models as shared resources. Why this focus? As you recall, during this morning's introduction, Rosa provided a historical background on how the goals for this workshop have been informed by multiple layers of stakeholder discussions over the past few years. So in order to focus today's discussion, we have considered input from various sources regarding the needs, priorities, and capabilities identified by California research stakeholders with regards to potential competency hubs. Shown here are responses collected through outreach to directors of past CIRM shared labs and a town hall meeting that we held early in 2021 that included providers and users of shareable resources. So as you can see in the graph, the results highlight that California research stakeholders identified validated stem cell models as a priority resource. This is ranked highest on a wish list for shareable resources. Next slide. And if I can just ask RLA, I don't see myself currently showing. It's Ilyas, just, but maybe that's just me. All right. This input led, thanks. So this input led to the focus of today's discussion on possible shared resources labs for stem cell-based modeling. And we want to discuss with this group of experts if this is a feasible and worthwhile approach. To further focus today's discussion, we will focus them on applications to the central nervous system as a use case. However, the outcomes will be applied broadly across cell types and organs. Next slide. So in preparation for the workshop discussion, we wanted to gain some additional insights into the current use of stem cell-based models and the current need for access to stem cell-based models. So we conducted a pre-workshop survey with a focus on CNS research, as you'll see. Next slide, please. So I'll quickly go over just the highlights of these results, as it's a little interesting for our discussions. So we asked survey participants whether they are interested in whether they are currently using stem cell based models in their lab. And you can see that more than half of the respondents said yes. And 60% of those, next please, and 60% of those who currently use stem cell based models get requests to share them with others. Next. Those who already share their models do so in various ways. They provide protocols and advice. They ship their model to other labs. They collaborate with others on the use of their model, or they train other researchers in their lab or through workshops. Next. We also asked the survey takers if they wanted access to stem cell based models. Next. And of those who not currently use stem cell based models, 40% of them said they were seeking access to models. And while while those who already use models, most were interested in access to additional models. So I think this is interesting. I think many of those who aren't currently using models are actually not active researchers necessarily, but you'll see, you can see that there's a great interest both amongst those who not currently seek models, but who not currently use models, but also those who already use models to get access to additional models. And then next, similar to the responses about sharing their models, those who seek access to models are interested in receiving protocols and advice, having the model shipped to them. They would like to collaborate with others on the use of a model, or they would like to be trained to create the model in their own lab. Next slide. We wanted to get an idea of the types of human stem cell based models currently used in California labs. Respondents indicated that they use 2D cultures, organoids, and some also indicating the use of 3D models. Next 2 clicks, please. You remember the survey was conducted in the context of CNS research, and here you can just see that many of the respondents provided generic neuron or brain specifications for the specific models they use. But you can see that researchers are also using stem cell-based models to study a variety of CNS cell types in brain subregions. Next slide. When asked what type of human stem cell-based model researchers are interested in obtaining from others, there was an interest, again, to use 2D cultures, organoids, and 3D models. And 2 more clicks, please. And there, again, was an interest in a variety of cell types and brain regions for modeling. Okay, next slide. So this is my last slide to kick off today's discussion. Today's discussion has the goal of helping CIRM understand if we were to support the creation of shared resources labs for stem cell-based modeling, is that a feasible thing to do at this time? What would be the best approaches to create a network of shared resources labs? And how should a network of shared resources labs be designed to help advance CIRM's goal of accelerating research? So for this portion, in a moment, when we stop sharing slides, if I could ask the discussants to please turn on your camera, if you don't mind. If I can keep the slide for a little longer, please. But also please stay muted unless you're speaking. In the interest of time, as mentioned earlier, I will not introduce each discussant. Please refer to the bios for each discussant in the workshop program for more information. And when you do so, you'll find out that this is indeed an incredibly amazing group of researchers and experts we've assembled for this workshop today. Thank you all for being here. So for this discussion, we've divided it into 4 sections to sort of guide our deliberations. I understand that things might be jumping around in various responses. But to now set the stage and frame the upcoming discussion, I'm going to go through these 4 guiding questions that we have. The first one is to reflect on what the biggest hurdles are to effective human stem cell-based modeling, and to think about how a network of shared resources labs could address them. So this is a question to a large extent about the state of the art and the scientific hurdles facing the field. And could CIRM, through the creation of shared resources labs, help with overcoming these hurdles? The second guiding question we'll get to is more of a practical nature, asking, what would it take to build a shared resources lab? What is feasible? What incentives would be needed for a researcher or a research group or an institution to want to provide a shared resources lab? Similar to what CIRM did back under Proposition 71, when we created the shared labs of that time. And then we're also interested in learning what it is that the wider research community needs in terms of access to stem cell-based models, and how would they like or would they need to access them. So sort of more practical questions around building shared resources labs. And then in the third portion of this discussion, we'll reflect on, having heard the previous discussion, we'll reflect on the overall feasibility of building a potential network of such shared resources labs for stem cell-based modeling and their utility in accelerating research. And then in the final section, we'll connect today's subject, stem cell-based modeling, with tomorrow's focus on a possible data infrastructure that CIRM might build. And we'll seek feedback from this group about the needs of experimentalists for support with collaborative data sharing and analysis environments. Okay, so yeah, you can stop sharing the slide now. And so as a matter of process, for this discussion, I will call on individual discussants to make comments, but I will also pay attention to raised hands. And we'll start by thinking about what are the biggest hurdles to effective human stem cell-based modeling, and how can a network of shared resources labs address them. I'd like to call on Steve Finkbeiner to get us started. I'm sure we'll touch on a variety of hurdles over the next half hour or so. And I'd like to first focus, just to get us started, on issues related to standardization and reproducibility, because that's often a hurdle that is mentioned when it comes to induced or pluripotent stem cell-based cell modeling. So Steve, if you wouldn't mind starting with your thoughts on that hurdle.
Thank you very much, Uta, and thank you, CIRM, for so thoughtfully trying to get the input from the community to match this great opportunity with kind of unmet needs. So one of the biggest hurdles in my mind is the variability, standardization, reproducibility issues, and we've been doing this disease modeling since 2009. We were part of one of the original consortia that NIH funded to develop disease models, and we've been working on it since. In 6 diseases, I think we've collected over 2,000 IPS lines, and we've used them for academic projects as well as large-scale screens with over a dozen industry partners. And I think based on that experience, it's really shown me that the still high level of variability in IPS lines, differentiation protocols, and assays is sufficiently high. It can make it difficult for one lab to replicate the findings of another, even when the written description of the methods being used is relatively detailed. And I think that's a major problem, because results that aren't robust and reproducible waste resources and form a weak foundation for really using these lines to develop knowledge of therapies. I think working on our own in several consortia, including one of my fellow panelists, Clive Svensson here, I think based on those experiences, we have found a number of sources of variability and approaches to mitigate them. I think some of the most important have been a shift to footprint-free methods of reprogramming, careful attention to regular and rigorous QC of the lines that are produced, gene correction in relevant situations, larger numbers of independent lines, automated liquid handling, optimized differentiation protocols, cell type specific reporters, I think high throughput analytical approaches and especially AI applications, and face-to-face training for members of other laboratories. I think all those things have made a difference. But that said, I still see huge opportunities with the new CIRM initiative and the development of the shared resources labs to tackle this problem, particularly in a systematic way. And I think it could substantially improve the quality and utility of iPSC disease modeling by increasing the rigor and reproducibility of the work, and by disseminating knowledge and supporting labs that access the work. Our experiences within a number of these consortia dedicated to modeling HD, ALS, PD, and autism has shown us how effective sort of a shared resource lab network model can be for providing lines, protocols, models, assays, or experimental results. And I think Dave Schaefer really nicely developed that in his earlier talk. And so depending on the needs of the collaborating labs, I think the shared resource lab network can help with training, best practices, and controls they need to do the work in their own lab or to have the work done with shared resource labs under highly controlled conditions that could help improve the strength and interpretability of the conclusions. And I think particularly the network aspect and being able to share data amongst the network and across the network could be an amazing engine for generating the sorts of data that would be especially amenable to artificial intelligence tools for analysis, creating both a depth and a richness that could lead to insights that would be difficult for any one lab to achieve on its own. And I think also could lead to the development of really cool new AI tools for analysis that could then be disseminated back to the laboratories for their use. And so I think this could really accelerate sort of the achievement of some of the missions that CIRM set up. So I think I've used up my nickel. I'll send it back to you, Uda. Thanks so much.
Thank you, Steve, for getting us going and covering sort of a range of approaches, that we would need to keep in mind that perhaps should be part of a network of shared resources labs. I see, Sergio, you wanted to chime in?
Yeah, so I mean, I very much echo Steve's terrific points, and these are certainly many of the sources. What I thought would be also useful to do is also think, I'm not sure whether that's the right term, but perhaps from a sociological point of view, why doing this type of experiment is so challenging, right? Because and that perhaps will help us think how we can address that as a community. Because one of the challenges that most of the protocols that we're using, especially three-dimensional protocols now, and certainly those that involve, for instance, organoids and further than even assembloids, are incredibly elaborate. They have not been truly standardized, and most of those experiments last hundreds of days. And so certainly the feedback that any new user in this field is receiving is not certainly on the right time frame, right? Like, just think about CRISPR, how easy it is to learn CRISPR because the results are easily quantifiable. It can be done even in a small lab. But these experiments are long, incredibly expensive. If done across enough number of lines, it certainly becomes prohibitive for many labs. And that has been, and I think Stephen very beautifully mentioned this, that the scale is so important for this experiment. And another point which I think is worth keeping in mind is that the field, especially for CNS, is becoming ever more multidisciplinary and ever more moving into neuroscience, so to speak, because the assays that we're envisioning now are ever more functional. And that really means patch clamping neurons, which is really a very difficult thing to do. There are not that many neuroscientists. The reality is for like patch clamping in this field. And I'm just mentioning one technique, but there's certainly imaging and perhaps the transcriptomic single cell profiling approaches have been ever more standardized. But to a large extent, most of the other assays where we are thinking that most of the phenotypes for disease are going to arise have not been implemented in human neurons very carefully. And that requires actually advanced expertise. And in fact, in our experience, my lab and the program that I lead here at Stanford has helped over 160 labs, one by one, to implement methods that we have developed. And it's incredibly difficult because every single lab has different set of questions and different set of issues to address. And they're long lasting, right? Because they're hitting multiple problems at multiple stages as they're thinking about their experimental design, about implementing various assays and so on and so forth. So I think it's important to take into account that we are facing challenges because of the intrinsic nature of the experiments that we are doing, which are long, elaborate, expensive, and require multidisciplinary assays. That would be my first point.
So yeah, thank you, Sergio. I think one aspect you are hitting on that is important to consider when trying to create these resources is, are they ready for broad sharing? So that's one of the things we want to understand from you all. And I know you use organoids, which you mature, for dozens, 100 days, etc. But there are also cell-based models that are much quicker differentiation and perhaps also already more standardized. And so one question I have for the group is when it comes to helping with the standardization and reproducibility issues that exist in the field, how shared resources labs, how a network of these core-like facilities could help with a standardization and reproducibility, both for simple models and for more complex models. Maybe I'll come back to you, Sergio, later. Maybe I'll call on Allison if he could maybe bring up, and I saw your hand, Ilyas, and I'll call on you next. But Allison, if you want to, let's go with Ilyas next since he has his hand up. Ilyas.
Thanks, Uta. Great to be part of this discussion. So what I would like to add is, so really what Sergio and Steve were saying, it's really hits exactly on the issues that we are dealing with and trying to solve here. So one thing to keep in mind is asking ourselves early on, so what is actually the goal of disease model? Is it really a basic biology question we are trying to tackle and compare to what we know from in vivo or clinical data? Or is it really drug development? Or is it really developing, based on your findings, a cell and gene therapy approach? So early on, this can be factored in and considered, because this will define how you basically cherry pick the assays that you want to develop, right? So the assay development is key to this approach. What endpoints are of interest? Mm-hmm. If you have the opportunity for high throughput, high content, this is obviously a great advantage. So in general, what we ask ourselves always is kind of how translational can we be developing these assays and trying to solve some of these underlying problems. So the other questions is always, can you use quantitative biology? Can you have dose response curves? Can you have internal validation and then quickly through maybe these collaborations and shared networks, can you ask a collaborator to reproduce your data? So we are very careful with actually making these correlations until it's really validated externally. So I think that's kind of something early on that needs to be kind of discussed and be on the table. But I think having these collaborative opportunities, also thinking about the scientific rigor that you can learn and implement based on what we know from drug discovery, right? How can you implement this for stem cell-based technologies could be really key for success. So the other areas of interest to keep in mind is really the question of numbers. Can you actually scale up? How many cell lines do you need to analyze to come to a very rigorous conclusion? And what we tried to do here is use automated technologies, for instance, so robotics can help. And another area of interest is the reagents that we use. We're trying to always justify why are we using a certain drug to manipulate the cell signaling pathways? So can we better justify? Can we have, dose response curves and independent validation essentially? So another area of interest is kind of revisiting some of the basic ways how we have been culturing cells. So we have, as an example, developed a small molecule cocktail, so published last year, that has really become the chemical foundation of what we are doing with essentially every step of working with iPS cells. So this could truly be a universally applicable platform for the field, as it really allows us to culture these cells now under stress-free conditions. And there's a paper we have in preparation where we actually demonstrate that the cell aggregation process at the very beginning of generating organoids is confounded by cell stress and cell death. So essentially, if you aggregate your cells and have lots of dead cells in your organoid, this problem basically you carry this basically for the next 60, 90 days and longer. So we see signs for stress even after long-term culture if you don't have the optimal start at the very beginning of cell aggregation, as an example. So the question again, is it a cell culture artifact, or is it really part of the underlying pathobiology? And what actually is compensation? What are these cells doing to compensate dealing with stress? So yeah, these are all very complex questions. I'm curious to hear what others have to say about this.
Yeah, thank you. One thing I maybe want to comment on is, you run, stem cell modeling laboratory, and you can make decisions about how to standardize, within this laboratory. And part of what we're trying to think through here is how to, the way I like to think about is herd all the California researchers into a network where they could, where they might not all use exactly the same methods. And of course, there's always the issue also of deciding on a particular method for standardization, but there's also the need for continued innovation. From what we're hearing, clearly this field is still evolving, and we perhaps don't want to fix certain standards necessarily. But I heard you also say an interesting other aspect, which is to use a network, and I think Steve Finkbeiner was also alluding to that, to use a network for doing similar experiments in different labs and combining the data and discovering what sticks in a way. So, but yeah, I'd like to get to, since I already warned him, Allison, to comment on, about his thinking around how to monitor the organoids and how that might help with, if not standardization, with having knowledge of what it is exactly that you've done. And if it looks like maybe Allison might not be here, but I see Bruce raising his hand.
Oh, okay, great. I had a different point. I'm going to just address that to some extent. I think that the first of all, a terrific topic and something that we've an issue that we've faced a lot. I think that the at Gladstone, I was the founder of the stem cell core, and that's been very, very valuable for teaching a lot of people how, and getting lots of programs, many of them CRM-funded, up and running, and it's been that has been tremendous. We look forward to the next step. One of the things that Uta, you're talking about, that struck me in the last comment was what you're trying to do is get the researchers to coordinate with each other. And one of the ways that this is done effectively with other large consortia, so for instance, we're in the somatic cell gene editing consortia for NIH, is essentially what they did is they put out an RFA that was where we funded, I think, like 20 different groups around the country. And then they followed it on in the first year, people get to know each other. Second year, though, right away, they start having funds specifically for coordination between collaborations, new collaborations between those groups. And so, for instance, in the genome editing thing, the people who are making new genome editors and people who are using them and trying to figure out assays for those, they fund collaborations between the two, and then ask for reports about this new collaboration. And so, even though the money is not actually huge in terms of what they fund, it does inspire many collaborations across within the group that wouldn't have occurred before. And just essentially, if you think about the whole project together, and think about the funds initially for funding individual groups, which are almost by definition related to each other because they're all responding to this RFA, and then have funding for collaborations that are inside California, I can see a lot of good that could come from that. So that's just a suggestion, and it certainly has worked for some of the NIH kind of consortia. So just a thought.
Mm-hmm. Yeah, I think collaboration will be, an important piece, and maybe those who so we have, of course, Dave Schaffer with us, who spoke this morning about his research lab and a shared lab, and then Bruce Yu were involved with the Gladstone lab, and then we also have Eileen Anderson, who ran the UCI shared lab, and so lessons learned from what could be done for labs to more collaborate, but also for researchers to collaborate, with the labs. Alison, sorry, it's Bruce.
Yeah, just the one thing was that the difference here was that you can have collaborations baked into the initial application, but it's really the more interesting one are the collaborations that come afterwards, because those are groups that didn't know each other before, almost by definition. So that just thought.
And that's something that we actually did with the Genomics initiative. So that would be potentially, something to use as lessons learned, is have the center that was funded do research projects, but then they were bringing in collaborative projects. So that's something to remember. Alison, sorry, I didn't see you earlier because I was on the wrong view.
Oh, good. Thanks, Uta. Yeah, so this is great. I do think that the collaboration will definitely help us to move forward. I also bring perhaps a more optimistic view. I see that during the years, several of these protocols, especially for the 2D cultures, a specific subtype of neurons are becoming more and more easy or reproducible. And the field kind of, go towards some of the simpler protocols. I think one good example is those MET inhibitors for the cortical neurons. I think most people here use that protocol. And I think the same thing would happen in 3D models as well. One thing that I do believe that it's important, and I see very few opportunities for researchers to do that, is to spend time on the optimization. I think Ilias is correct that this can be done. You can either save time, money on reagents if you really optimize. I love the work that he did with iPSCs. I think that's possible to do with several of these cell types, even 3D cultures. The Cortical Organoid protocol that we developed to create networks is a clear example. It took 4 years to optimize that, but finally you can have these networks functioning inside an organoid. So that we couldn't see that with any other protocol that we try, but requires this testing. So I'm so glad that David Pashinson is here, because we kind of use the NIMH resources to kind of optimize the protocol. I know Serm has the tools and technology that perhaps could be a venue to do that, but I think the level of support to get to the stage where we are needs to be significantly higher on the funding side. And that might be the first step. Once you fully optimize something, then you move forward with a more larger distribution. So I think it can be done. I think we collectively we can achieve better protocols that are more reproducible, that many people would end up using.
Mm-hmm. And if you, but it also monitoring, what your model is doing and having that information to go with the result.
That's another important thing. I mean, because you might tweak the protocol to reach what you want. Sometimes, I mean, you want just more activity and you don't care about the cell types that are there. Sometimes you just want more representation of the cell types. You can get that, but then, I mean, there is a cost that you pay by having perhaps less organization. That's a clear example between guided and non-guided organoids. So you can choose the ones depending on your biological question. So you are absolutely right. I mean, we have to be thinking about the readouts before we do that.
Thank you. So I count 4 Davids on this panel, but I see David Panchisian has his hand raised.
Yeah, thanks. Lots of great points being brought up. And I want to maybe first start with to touch on Bruce's comment, because I was involved in the somatic mosaicism network. So doing these types of consortia where you incorporate a common experiment works really well when people are where the outcome measures are very similar, and the ask is very circumscribed. Where it becomes a little more challenging is where you have a lot of open-ended, where you have an initiative that's relatively open-ended, and where there are potentially a lot of different directions going on. We had the NCR-CRG, which is a public-private partnership to optimize protocols, and also Convergent Neuroscience, where they were all using iPSC-based approaches in addition to other things, but they tended to be rather divergent in either the disease orientation or the suite of approaches. And there you kind of have to pick and choose where there are opportunities to collaborate outside of the original applications. So it really depends on how you want to structure the organization of the initiative. One thing I do want to raise, which other people have touched on, but I kind of want to give it a slightly different flavor, which is this issue of achieving predictive and explanatory value. And I think this is critical, but it's really challenging to do because most diseases are complex, right? Either they have multifactorial contributions to risk, or they have a highly distributed functional feature of the disorder. And representing this in human cell-based assays is challenging because they're inherently reductionistic, right? And so one of the ways that we've approached it is thinking about it in terms of a suite of fit-for-purpose assays where each incorporates some discrete predictive or explanatory feature of the disorder. But getting to that is tricky, specifically because cell-based assays don't have an intrinsic ground truth. Well, they do, but it's genetics, and that's why people use it. But in terms of phenotypic output measures, they rest largely on how well the assays map against some reference data. And this can take the form of clinical phenotyping, or it can take the form of some reasonable surrogate. We use postmortem tissue genomic annotation, for example, quite extensively. And model organisms, of course, are used, but we know all about their limitations, especially when they come to things like psychiatric disorders. And so in the context of your question, I think one way of doing this is having a network of labs that have specialized expertise in specific approaches, but they work together on the same data production pipelines, and they can do cross-paradigm analysis. And this can be quite powerful if they're working together on the same disorder. So having some sort of nucleating concept behind the groups working together so that there is sort of conceptual cohesion so that they can work together and do these sort of common experiments, I think could be quite powerful.
Thank you for that. Yeah, that sounds very good insight. And I also want to just acknowledge that you've emphasized the need for better understanding the predictive value or predictive power of these models. And I've talked with actually all of you before this meeting, and this came up in various discussions. And so thinking through how, as a group of not only the shared resources, potential shared resources labs, but then the California researchers collaborating with them, how we can funnel some of these efforts into collecting that data and improving our understanding of how predictive these models are. Eileen.
So I think these are all great comments. And I think, as Ilyas mentioned a few minutes ago, something critical to consider is what these models are going to be used for, right? And therefore kind of leading into this issue of whether they have predictive validity. And in that sense, I think it's important to pause and think about the pieces that are still missing from those models, even, as far as they've developed so far, and that there is some consistency emerging. They often lack mechanical forces, which we know are really important in terms of fate and migration and differentiation and how cells behave. They generally lack any integration with the immune system or with immune factors, with blood flow. So I think it's important to just pause and step back and think also about what role there is to be played in terms of the next generation model development, because some of that is already ongoing, but it is going to come. I don't think that we're going to be able to stand back and say we have the right model for every disease, for every condition, for every drug screening. And that has to be a part of the development activities as well, and potentially what functions a core can serve.
Yeah, thank you. Again, also bringing us back to sort of the conundrum of trying to figure out, should we focus on well-established, reasonably well-standardized models, because we want this reproducibility versus keeping innovating and creating these more complex and more realistic models. And again, yeah, I really appreciate you all helping us think through how both perhaps could be enabled by a network of shared labs. Steve.
Yeah, I just wanted to underscore, I think the question of how do we develop more predictive models is such an important one, but at the same time, such a hard one. I think David offers one idea. Let's try to think of all the different aspects of the disease, kind of getting a face validity around certain assays that replicate different parts of the disease. And I think that is a well-established general approach to this problem. And then Eileen mentioned the issue of complexity, sort of adding other dimensions to the models that don't exist, with the idea of kind of hoping that that will make them more physiologically relevant. But it's so challenging because every time we add complexity to a model, that tends to also add variability, and that also can undermine then the sensitivity of those assays for the sort of work we want to do, make them also more difficult to disseminate and share. I do think, though, that one of the most exciting aspects of working on especially patient-derived iPS cells is that we do have clinical information for a lot of these patients. And what I really would love to have are compounds that work in the disease that we could then use to reverse engineer these pipelines and figure out which models, which conditions, which things actually would genuinely have predictive value. But we don't have that. And yet I do think, and this is kind of the approach that we've been taking, I think there are opportunities for really advanced computational approaches like AI to be able to, in a more holistic way, look at the phenotypes that we get out of our cells and ask, what are the things in a dish that we can measure that at least most predict clinical features of the patients from whom those cells came? And I think that may be an interesting avenue as well, to try to at least help us focus what are the things where we can draw the strongest connections. And I think it's at least worth thinking about in terms of answering this really important question: how do we make our models more predictive?
Thank you, Steve. Sergio.
Also related to this, and I think to the question that you posed earlier, Uta, whether we are, are we ready, or is this the right time? I mean, whether we're ready or not, this is happening. I mean, I think everybody's seeing around that there are more and more labs that are trying to do this. Whether it's like prompted by curiosity, by students want to do it, by the mirage of what you could do perhaps with 3D cultures, by funders, reviewers, all, a lot of labs are moving into this field. And, standardizing and working on standardizing at least some aspects before reaching too much complexity, I think it's essential, and I think it's worth keeping in mind. That was my brief comment.
Thank you. Ruth.
Thank you so much. And we're very grateful to CIRM because we were a recipient of a CIRM grant to actually facilitate acquisition of tissue from a large cohort of individuals with autism spectrum disorder and also typical developing controls. And I think the point that was just raised by Stephen and alluded to by David is that, of course, the data in is very important. So the clinical phenotyping is pretty much where I reside, and that's essential. So there was a comment earlier about the controls, the 300 controls across the extant registry. And of course, there's variability in what's an appropriate control for which disorder. For example, in our own typical developing controls, we had a social communication questionnaire to assure that they didn't fall into some twilight region between autism and autism spectrum broader phenotypes, so that they would really be clean controls, yet that was not acquired on every control. And so I think there's an opportunity to refine our phenotypes, perhaps, to go back and obtain more objective measures. We've been very fortunate to be working with many collaborators, David Amaral up at UC Davis, also Sergio Pasca, and we had the opportunity to bring back individuals for neuroimaging, which is, has its own heterogeneity and variability, but is a more defined objective measure, particularly for particular phenotypes of ASD, such as megalencephaly. So I think we cannot potentially avoid not having to at least collaborate together around controls and other clinical phenotype definitions, even within the extant registry. That said, we had a tremendous amount of collaboration in that initiative by CIRM. CIRM paved the way for such uniform procedures from consenting through phenotyping. We worked very closely with the UCSD site and Joseph Gleason down there to make sure that we were getting the same information on our individuals that we were recruiting with ASD. So I think there are opportunities already that exist where we could build on those to actually forge this kind of collaborative initiative, not just at the cellular model or output, but also at the input point where we're actually clearly defining the phenotypes we have. And the one thing I would like to add, there was a wonderful point made by Eileen about the future, and we have to keep an eye to the future. So models where we look at how neurons are interacting with different components of physiology, such as immune function, is a tremendous opportunity. But I think we have an opportunity there already. There is a hypothesis around ASD of cardiac dysfunction. We have excellent collaborations here on cellular on cardiomyocyte models that CIRM also contributed to the development of. And we could do some of these in parallel from the same individuals that might be very useful without having to wait for more sophisticated integration of different physiological cellular models in the dish.
Yeah, thank you. Yeah, and also for bringing up indeed the the phenotyping of the donors that goes, or clinical phenotyping of the donors that has to go into these analyses. And I know David Amaral has a lot to say about that. So David.
Yeah, thank you, Uda. And I just want to follow up on what Ruth said. I thought it was going to come a little bit later, but I think it's a good time to mention it now. And we've been using iPSC strategies as part of an NIH-funded Autism Center of Excellence. And the issue that we're trying to address is the mechanistic etiology of various forms of autism. So Ruth touched on this. We've demonstrated that about 15% of boys with autism have megalencephaly or enlarged brains.
but the neurobiology leading to megalencephaly is unknown. So, but we've known from highly phenotyping these children that this form of autism has the poorest prognosis for cognitive development and reduction of autistic symptoms. So this finding of these kids with the big brains and autism has come in the context of a much larger study that now involves 600 families. And we're collaborating with Ruth and Joaquin Hallmeyer and Sundari Chetty, developing iPSC cells from these kids to be able to study glial cells and neurons from the children in the study. Can't go into it now, but we've developed data indicating really quite interesting different transcriptomic profiles from cells of the megalencephalic kids relative to kids with autism who don't have megalencephaly. So we're already seeing a differentiation. So I think what's been most beneficial to this research is that the children are very highly phenotyped by the team at the UC Davis Mind Institute. This ranges all the way from medical evaluation to comprehensive behavioral assessment, including MRIs and EEG studies. And what's interesting is that our team at the Mind has been able to recruit kids at all severity levels in autism. Generally speaking, people that are studied who have autism are the highly functioning, highly compliant individuals, but we have children in our studies that have IQs down to 20 who have no language, who have self-injurious behaviors. So we're really looking at the full spectrum of autism spectrum disorder. And I think this allows us then to come up with specific hypotheses to test using iPSC cell lines from the highly phenotyped children. And so I guess my point that I was going to make later is that a very important component of future CIRM-funded networks will be partners in the networks that are proficient not only at recruiting and evaluating patient populations of interest, but this is particularly important when dealing with neurodevelopmental disorders such as autism, where obtaining reliable behavioral and biological data can be extremely challenging. So I wanted to echo what Ruth had said, that it's great to have large numbers of iPS cell lines, but if you don't know about the clinical characteristics, behavioral characteristics of those cell lines, I think you've missed a huge opportunity. And what we would love and what we've been doing with the team at Stanford is to actually have active discussions about the phenotypic characteristics of those children as they're looking at the cell lines in the cultures. So thank you.
Yeah, thank you, David. And it does bring up a little also, I think, the fact that there are amazing resources that already exist, like the one you just described. And that part of what we need to think through is how to bring them into the fold when designing, let's say, these shared labs, these shared resources labs, instead of recreating what you just described. How can we bring in various other resources and leverage them, for something else that then would be achieved by this network of shared labs? And I'll call on Ru in a moment to talk a little bit about, for instance, the resources that the Allen Institute has that might be of interest there. But Matt has his hand up, and
I just wanted to briefly follow up on what Ruth and David were talking about, and I think this is an important point that we need to realize that we can't assume that these patient profiles, their diagnosis, are static. So, for example, UCI contributed quite a lot of samples for the Alzheimer's cases, some of which started as controls. Some were mild cognitive impairment, some are Alzheimer's. Those patients are continuing to be followed, and many of them are converting and changing. And so I think we need the mechanism to continue to update our information about these patients and how they're changing. And likewise, we're doing more endpoints. We're assessing new biomarkers, new PET scanning data is becoming available for those patients. And so making sure that people realize and add those resources to the availability. I think we need to keep, even though there's this great resource here, we need to update it and continue to update it, because some of those diagnoses are going to change from control to Alzheimer's here.
Absolutely. Thank you. Clive.
Yeah, just to follow on from that, the control issue, we did create 30 lines from patients who reached 82 in Scotland, for the Lothian cohort, where they were perfectly healthy cognitively, were in a couple of categories, but all cognitively intact. They didn't have diabetes, didn't have Alzheimer's, have Parkinson's, and created 30 lines from those patients. So it's another strategy, taking patients who have made it. And in Scotland, if you can get that far, it's a big deal with all the haggis and the beer and everything else. That's another strategy, is to create a resource of patient samples with really well-defined characteristics, because controls are really missing in a lot of these studies. And a lot of people, yeah, you can get, we've done 1,000 IPS lines from patients with ALS, but trying to get the controls to match them. And when you start doing informatics studies and big data, you need balanced groups, and the controls, I think, are critical. So Serm created a really wealth of control IPS lines that are going to be very valuable.
Yeah, thank you. And yeah, so this, sort of this discussion has highlighted, although, I really want to focus it on the actual differentiated models, that we, as a discussion subject for today, how to share that. But I think what you all are highlighting is that even, obviously the input material, which is going to be mostly human-induced pluripotent stem cell lines from people with various conditions, is still, a resource that needs to be well studied and characterized and considered, as they're, coming into this future of shared cell models. Mike.
Yeah, I agree with everything said. To me, it's a no-brainer. You want to build this shared resource and form the network. It's a high priority line, and you start out with the most specialized system. You bring in all the stakeholders who, because nobody really wants to do anybody else's protocol, but you got to get them all in the same room so that they ultimately will converge. Just one thing for thinking about in the future, it's not, it's great to bring in all these lines from patients. Totally agree. The better phenotype they are, the better the resource will be. And I love the idea, we're generating data for all different groups, would be generating data, which would enhance utility. I do think one thing to keep in mind is that it'll have most effectiveness if you can spread these lines out to as many labs as possible. So make sure that it doesn't just stay within a closed network. And I know that seems obvious, but it's not always implemented as well as you'd like to see. To make sure that, quite frankly, you want every researcher in California to have access to these resources. Mm-hmm. The other thing I think to think about, which I haven't heard said yet, is you're not just building a resource with what we have, but if you think about it, there's probably, we want, for example, libraries of CRISPR mutants in these lines. You heard about the village experiment. So you want to have resources around each of these things with that, so people can do screens. If you build a large synthetic type of resource that can be used for all kinds of different experiments, comes back to Elias's comment. When you think about the end user, there really are a lot of different end users, and there's no question having CRISPR lines minimally around, a lot of the standard models, but definitely possibly around all knockout genes would be quite powerful. So you want to be building really generally, high utility resources.
Mm-hmm. Yeah, thanks, Mike. I actually want to highlight to everybody that Mike was the PI for the Genomics Initiative. And when he just spoke about, bringing everybody into the room, one of the features of implementing and that initiative was that this large group of not only the center itself, but all the collaborators would get together, I think, once a year to talk about progress and solve problems together. And I think a lot of collaborations came out of that. And so maybe I just wanted to highlight that as a means, for networking any sort of shared resources that we might create. And then thanks, Mike, also for, just, confirming that and that's what we're trying to do as we CIRM think about such shared resources, is how do we get them to as many people as possible? And because I think ultimately the more people will have their hands on these resources and study them, the more standardized, or the better we understand what all the results will mean that come from many people working with these lines. Ash.
Yeah, I'll kind of echo what a lot of people here have said. one of the things that I think a lot about, is really, how to make things robust and standardized. And I think, one of the challenges that I've heard here is, everybody has their different protocol for doing a differentiation. It's kind of like cooking. Even if you think the protocol is the same, it's different. Kind of dual SMAD inhibition is not dual SMAD inhibition from one lab to the next. And I think really focusing on how to kind of cut through that, taking a reductionist view. And maybe one of the things that could be done is a centralized place to make progenitors, or to work on, for instance, some of the transcription factor protocols, which are not perfect, but are much more robust, across multiple lines. So instead of kind of spreading that out, having a central place that focuses on kind of generating progenitors for all of the lines, generating large scale banks, as Mike said, in terms of CRISPR knockouts or CRISPR perturbations. I know that the Indy project that Michael Ward is organizing is really kind of moving forward on that. And so really kind of thinking about what are the standardized things that then could be then distributed, kind of to then deeply phenotype. And I really like what Steve Finkbeiner was saying about kind of using machine learning to identify phenotypes. I very much embrace that. But if we could have a standardized central resource of kind of progenitors, then you could then ship those out, and it becomes, what is the phenotyping that you're doing on those? How deep can you go on phenotyping, and what's the right phenotyping that you need to do to make a predictive model, both in a human and both in a cell model? And connecting those up, to me, is something that I spend a lot of time thinking about. How do you actually go from this model that you've built in a dish to really know that it's predictive? And I think that's like the barrier that we face. All these different labs doing different things, and nobody can reproduce this phenotype, kind of because it's been done in an organoid or it's been done in a transcription factor induced protocol. I think really standardizing that as a central thing, and then having deeply phenotyping those, kind of as deep as we can go, I think is going to be a huge step forward. So that would be my kind of synthesis of what I've heard. And I really would love to be part of that. The other thing that, we haven't talked about is kind of industry academia partnerships, and would love to think about how we do that. I saw that a lot more when I was in Boston, when I was at Novartis, and have not been involved in that, kind of in California. So just a little bit of a love to forge ahead and actually build some more of those partnerships here in California.
Oh, great. Yeah, that's a very good point, actually. And I guess I'm not surprised you were excited about Steve Finkbeiner mentioning machine learning, but that's another thing to consider. How, is it like a large scale? I'm assuming for like in Citro does, and I think Steve's work also is highly automated and uses a lot of lines and brings in a lot of data, and which is then amenable to machine learning approaches for learning, gaining knowledge from all that, versus distributing smaller groups of models to many different labs, doing different works. It's something for CIRM to consider whether we can, I would love to be able to do both, of course, but we'll see what comes out of all this. I'm going to call on George. I see, Ilias, you have your hand raised, but I'll call on some folks who haven't commented yet.
Thank you, Yuda. And I just want to thank you and Rosa and CIRM for putting together this incredible group of folks. And I just find this conversation so inspiring. I want to offer a comment that is a different approach from what's been discussed before, which may or may not be popular, but I'll be bold and say it anyway. I think that there's no doubt in the upside of investigating the pathophysiology of very specific diseases and specifically for reproducibility and standardization reasons. Another perspective is that there's a huge upside potential that could be gained from moving away from disease-specific modeling to a research and a collaborative strategy that has the ability to elucidate psychobiological processes that increase risk for chronic disease in general. And some of you may be aware that recently the state of California has put a lot of money into increasing awareness around screening for childhood adversity, for example. And the reason for that is that these childhood stressors increase risk for a lot of different disorders, like a 1-point-fold increase in risk for diabetes, mortality related to diabetes, 2-fold increase in mortality risk from heart disease and stroke, and a 2.3 or so increase in mortality risk from cancer. And those are just the chronic diseases. If you focus on the psychiatric and neurodegenerative disorders, it's much higher. So there's, a broad question about how do these stress-related effects occur? What's the neurobiology of stress-related disorders? And this all, I think, has the potential to lead to new diagnostics and therapeutics that could be used to reduce negative stress-related effects. The other, and I don't think that this takes away from any of the standardization and reproducibility issues, because these stress-related pathways have been well described, but they haven't necessarily been investigated with cell-based models and with the types of approaches that CIRM could be able to support. So one thing that I would just like to offer is some goal or window toward trying to use or harness the tools, resources, and knowledge that we have in this room and apply it to stress-related biology in a way that could really lead to breakthroughs in diagnostics and therapeutics around this common risk factor for a lot of chronic mental and physical health disorders.
Yeah, thank you, George. In terms of the diseases that will be modeled if this shared network of shared resource labs, were to become reality, the hope, I think, for CIRM would be, to bring in a lot of different opportunities for modeling different diseases, but I'm very glad you brought up your interest, and I think, I think you already work on depression using iPS cell-based models, but bringing in bringing stress, which is an important neurological or problem, would be fabulous, and I think, and the fact that the California Surgeon General's office has an interest, in this particular subject, and I also know many others at CIRM, at the board, are very interested in bringing these psychiatric conditions into the fold, and so part of, and we'll come to that maybe a little later, more deeply into the discussion, is to understand how to bring in researchers who study humans and have hypotheses about some neurological problem, but do not currently use wet lab work, let's say, or animal modelers, who have hypotheses about how the brain works or how a disease works, and how to bring them in through these shared resources. That would be definitely one of our goals, is to bring people not currently using human-based cell models into this research community.
I'll just mention very briefly that there is a ground truth in this area of research as well, which is that you bring human participants into the lab, you stress them out for 5 or 10 minutes, you get their stress reactivity profile, and then you validate your cell-based in vitro models against that real-world stress reactivity model. So I know in this area, we think of the ground truth as predicting to symptoms, which I think has a huge, which is very important. But in the stress world, we can also validate against a ground truth, which is people's naturalistic response to stress, which I think has implications for many of these disorders we're discussing. Thank you.
Thank you. Thank you for that. Yeah, interesting. Arnold, what are your thoughts on all this?
Yes, thank you for raising that issue. Well, I'm a little bit confused. I have to admit, we've heard a lot of highlights of the problems with some of these resources. The fact that organoid models and stem cell derived models generally have a lot of variability. They're inherently variable and they're just very complicated. And the problem with trying to reduce them to something that's manageable for everyone is that everyone's needs are also very variable. Some people want certain regional organoids. For some people, a sort of a generic progenitor neural progenitor might suffice. Other people need very specific types of progenitors or different cell diversity than the next person. Very difficult to come up with a protocol that would be standard that everybody would like. But
it does seem, if we go back to the two resources CIRM had generated in the past, we heard very nicely from David Schaefer about how successful resources were that provided shared equipment, resources that everybody, or at least individual labs, may not be able to afford, but need to have access to. And there's some new technologies coming down the road that I think very nicely fit that bill, where it's going to be important to have a core facility that multiple users can access, that will really relieve a roadblock or really drive research forward. And those are the more standard core facilities we're all familiar with. I can think of spatial transcriptomics, of multi-electrode recording, of some physiological phenotype marker that multiple labs might all want to use, despite working on different diseases and different tissues. Mm. So that kind of resource I can see CIRM supporting, and that would be terrific. And then the other is, of course, the IPS core that you described, where multiple different lines have been generated, and we've heard already that there are some shortcomings in terms of the lines that people have available. It would be wonderful for that starting material to be available, highly annotated, whole genome sequenced, from well-characterized clinical samples, and already generated into pluripotent cell lines using a standard protocol. That starting material that multiple different labs with different interests could share, to me, that also makes a lot of sense. And so I see these two kinds of resources that CIRM could develop, and they're very similar to the ones they did in the first round of CIRM. Mm-hmm. But I still think they're very, very useful, and mm-hmm. I would like to see that happen. Mm-hmm. But I'm just worried that standardizing protocols at this stage could stifle innovation, and mm-hmm. most of these in vitro platforms are still evolving, and there's a huge amount of work to be done. People are doing it, but there's nothing quite standard in my view right now, and I think mm-hmm. it could be a mistake to try to force some sort of standard protocol that everybody accepts at this stage.
Mm-hmm. Yeah, thanks, Arnold. I'd like to just encourage thinking about whether a network of shared studies on cell models, as opposed to everybody use the same protocol, but networking, let's say even everybody who has an interest in, let's say, Alzheimer's disease, everybody uses a different model, but how can, could a network of shared, of sharing of some sort, including detailed data perhaps, and with very strong metadata as to, what went into each experiment, and then, maybe putting machine learning on top of all of that, could that be a way to try and make a little bit more sense, of a complex field that you described, as opposed to, like you said, as opposed to deciding right now, this is the protocol and everybody do it that way. But yeah, thank you for that perspective. Sergio.
Yeah, but I think it's not about like us or the community deciding what is the protocol to use. I think it's to a large extent about setting a set of like quality controls that mm-hmm. would be applicable across multiple protocols. And I agree with Arnold that we should not like decide, oh, this is the protocol to use, because that would certainly stop some of the innovations. But there are certain quality controls that could be implemented. It's still surprising how very few labs are doing, for instance, any sort of like genomic analysis of their lines, like beyond just like the classic karyotyping that, can discover much, right? Or just across like neural differentiation, that there are like certain steps where certain markers should be checked, or, some of these practices, and also building on like Arnold's first argument about, the questions and the needs and the protocols. Like what I've seen quite often, especially in the last, year or so, is that there's a lot of misinformation, not disinformation, but misinformation about the protocol. We would often have somebody contacting us saying like, could we implement your, perhaps the most complicated assembloid model in our lab? And when I ask like, what is it for? Then they ask, oh, we studied this gene that is expressed in progenitor, so we want to find, so people are very often like not familiar with what are the available tools today for asking specific questions. And sometimes it may be a transcription factor-based differentiation, other times may be a monolayer differentiation, another time may be a transplantation model. But I think even that effort of conveying somehow in a concerted way what are the available tools for asking specific questions, I think that will also be incredibly useful.
Yeah, thank you. Mm-hmm.
So,
Clive, did you want to?
Yeah, I'm just going to riff off what Arnold was talking about. And I think he's made a good point that it's very early to do this in between part. So I see the IPS at the top, and we can do that pretty well, and we can actually standardize it. And in the embryonic stem cell world, I was in Wisconsin, and Jamie discovered H9, H1. Went a long way because everybody at least was using H9 or H1. In fact, we knew H9 made neural better, H1 made heart better. We all kind of knew these two lines. They were our friends, right? IPS is just like we just don't know, and it's don't know the genetic integrity of a lot of the lines when they start the differentiation. So I'm going to suggest sort of straw man for a resource would be some sort of IPS core facility that has, whether it's a Serm one or you could create your own one, of a select, say, 20 controls, 20 Alzheimer's, 20 Parkinson's, 20 ALS, or some number that's been really well characterized. And then in the middle, you've got labs, right, like Sergio's, and that do great differentiation. So you fund different pockets of labs that do great stuff. And then at the bottom, you've got your data portal, which should be common to everybody. So the same input from a set of IPS lines goes into different labs. They do their cool stuff. They distribute the data, and a lot of them will be focusing on the same thing. So if we had a set of Alzheimer lines with certain ApoE types that fed into that, out the other end comes the kind of predictive phenotypes they'd seen. That goes into the database at the bottom, you'll talk about tomorrow, and that gets widely available. So that way you can at least link the data that comes out with a line that went in, and then the magic kind of happens in the middle. Because we don't know, to validate a process, this came up earlier on, it may be that just a simple differentiation is enough to validate something in epilepsy that would give you a good prediction whether a drug would work in a kid with epilepsy. Or you might need to do a 300-day protocol, organ chip, vascular immune system. The truth is we don't know yet. So you only want to go as far as you get a validated technique. And this might really energize that if we standardize the beginning process and allow common access at the outset.
Thank you, Clive. Yeah, that it does fit sort of the idea, the vision, of how a network might function. Before I get to Ilyas, I did want to mention that from the attendees, a suggestion has come in, to also consider, and I'll just throw it out there, if it would make sense, instead of building a lot of small or supporting or creating a lot of small shared labs that are networked in a way, why not build one big infrastructure that is that has the opportunity to basically bring in, I guess, the power of scale, something like that in situ can do by deciding to focus on the disease, etc. So I know this might not be the best model for CIRM as we try to really serve as many researchers as possible with our competency hubs, but I just wanted to throw that out there as an idea. And I guess Ilyas also has the opportunity to run his big lab, in the way he sees it works best. Ilyas, you had your hand raised, but you're talking right now and I can't hear.
Yeah, so the key question for me is always, so what is really the end goal, right? So are we trying to find new disease signatures, right? So maybe in some psychiatric diseases, we don't have enough information. Now you have a cellular model and you want to connect this to your genetic data. So that's great. I mean, there could be an independent effort supporting that. But the question for me is kind of, if you want to help patients, right, and you don't have the next 50 years for this, right? So you have the next 5 to 10 years. So you have to come with a different approach. And in this case, if you already have information on the disease or disease phenotypes, like protein aggregation, oxidative stress, I mean, you already know what you will be looking at, right? So you could specifically model this. And this would kind of entail a different approach, especially if you think about drug development, right? So you could really have more targeted efforts. You already know the disease phenotype you want to tackle. But at the same time, I agree. I mean, you have to keep, innovation going. So you don't want to force everyone to do the same protocol, but nevertheless, I think you can decide and have consensus on the basics. How do we culture iPS cells? So even that is until now so spotty in the community. Some people still use mouse feeders, right? So because that was the traditional way of doing things, and it's kind of difficult for some folks to step away from that. But we know at this point that it has really tremendous implications in terms of translation. If you really don't use your chemically defined conditions at the very beginning, have the standards established. So the rigor and having kind of, a guiding principle that could lead to, let's say, CIRM approved protocols, right? It could be standard operating procedures that everyone can agree to, but not just forcing people to agree, but to really convince them, right? So to say, look, test this in your lab. So this is step by step the approach. And if they don't agree, or they say, no, we don't arrive at the same conclusion, then you will have additional discussions around this, or additional experiments to come to this consensus. So I think it should be kind of a fluid model of developing these protocols, testing them. Of course, you will keep improving on these, but nevertheless, you can have standard operating procedures, simply growing the cells in a certain way. But again, also being open-minded and following the data and have this kind of productive, forward-looking approach. But ultimately for us, it's important that we develop technologies that we can translate, that we can use for drug discovery, for cell therapy, etc. So that's for us always the key question. So if you look at cancer cell lines, right? So uncontrolled cell growth, you're modeling, you have a disease model for cancer, but we haven't really, cured cancer at this point, right? So it is more complicated than that. So just having a disease modeled in a dish doesn't necessarily really translate into a new therapy that can, save many lives. So essentially, kind of having a more focused discussion on what exactly do we mean by disease modeling, and what is the goal that we want to reach, right? Yeah.
So yeah, Ilya, so I mean, from what you're describing, I can also imagine this could be part of how a network could function, is where different shared labs and different collaborators with those shared labs, do the research they do, but I think one important function of a network could be for the groups to come together on a regular basis and check in on their protocols and their findings. And again, I might have a hard time imagining that we could convince all CIRM researchers to use the same protocol, but but be good about describing what it is that they exactly have done. And then maybe Mike Snyder can chime in. I'm going to call on Vijay next. The Genomics initiative did develop, for instance, what they called a minimal information about a stem cell experiment. And I think all the data that came into the data warehouse reported, on all and it was a huge spreadsheet. So I don't know, if that's a barrier, but I think in the end, the data, that were the outcomes from all these experiments were associated with knowledge around what went into the experiment, what protocol was used, etc. So that, so even if not everybody will use the same protocol, at least maybe we should make sure we know what it was that people used. Vijay, I'm going to call on you in a moment. I just wanted to well, first state that my vision for these separate sections for this discussion today, I have abandoned, and we are addressing various aspects that I wanted to come to later, sort of intermix, and it's not surprising since these subjects are indeed interrelated. Just to make that point, I also want to come back to the idea of leveraging existing resources, and thank you. Many of you have already, certainly talked about previous CIRM resources, but also I'd like to talk a little bit about other resources that exist that could be brought into the fold. And I also want to come back to, we have 3 shared labs directors, Prop 71 shared lab directors or former directors here, to also talk about how those Prop 70, what we here at CIRM call the Prop 71 shared labs, how they would fit into what we're talking about. But Viji, I'll call on you next. Are you there? We can't hear, I can't see you anymore since you lowered your hand. You might be muted. Viji, are you? We'll come back to you in a moment. Mike.
Well, I was just going to comment on the standardization and then give you some of my thoughts on this. On the standardization, sure, you need standardized metadata around the samples and the patients and all that, the front end part, I would say, and then you need standardization around the back end. It does take time to work that out, but it is all doable because everybody has a vested interest in building something they can, work across different labs for us. So I think it will happen. It just takes a little time. I do think as far as I don't know what you're thinking from the implementation side. Mm-hmm. I guess I envision this as a smallish number of groups that would produce large amounts of material that would go out to lots of people. And material could be finalized products that people would analyze, but could also be cells that people would use in their situations in their particular end use as well. Mm-hmm. So I think if you have way too many people, it's like herding cats. You get nothing done. So in my mind, there's a productivity and output that diminishes if you have way too many people, but you want to get these reagents out to as many people as possible. So from the shared resource side of it, I don't think you want 50 different labs all making, 50 labs can have some input initially, but the number of labs that would actually build these resources and get them out to the community, I would have thought would have been much smaller. I don't know what others were thinking, and how large N is open for debate. But I wouldn't think you want more than half a dozen labs. Maybe I'm wrong because I'm not a CNS expert, but making these things, that's my 2 cents.
Yeah, Mike, when I was referring to this larger, bringing everybody into the fold, that was the aspect of who will be receiving these resources.
Yeah, okay, that's great. Yeah, I mean, I think that did work for the genomics core, just to add to what you said, and you said this already, that we did have groups initially involved in this, but the collaborative projects really expanded the network. And then I think it worked well. People got pretty excited, set up all kinds of collaborations, and also it really helped build the standardization as well, because virtually all the stakeholders seem to be at the meetings.
Yeah, okay, thank you, Mike. Stuart.
Great, well, yeah, I'm actually in the second group, but since you opened it up, let me
yes.
take my hand down.
Yes.
Okay. So first of all, I totally agree with Arnold and Sergey, but I have a slightly different take, although I think it's what they're both saying, is that quality control is so important here because, it's garbage in, garbage out. So I think Clive said this too. And I think if that as a first principle could be served here, it would go a long way. It's almost like all of us refereeing each other before it ever gets to that point, which is the way it should be in a collaborative atmosphere. So yeah, I think that would be so important, at least at the IPS stage and sharing protocols. So I wanted to say what I thought scripts could do to this piece and some of the more functional phenotypes that I think are so important and closer to the disease. So as Leonard Mookie's group and my group has shown,
let's take Alzheimer's disease as an example. When Arnold will tell you why I still owe him nights on call. We were residents together, and I was the one who got sick. But Arnold, you remember, we were taught that in Alzheimer's disease, they never had hyperelectrical activity or seizures, and we were taught wrong, okay? Because many Alzheimer patients we now know have a hyperelectrical state and subclinical seizures. And Leonard Mookie deserves real credit in his group for showing this in mouse models. But we've reproduced that in cerebral organoids, and we can use that as a functional screen. And so I think it's so important not only to use all of our omic approaches, which is critical, but a functional characterization that relates to the real disease. And so we have at Scripps essentially high throughput patch clamp MEA calcium imaging system, coupled to our fantastic chemists that I would like to offer to all of you through the Caliber drug screening. I've talked to Pete Schultz about this as a drug screening initiative. And, we'd be honored to collaborate with people and help them with that. We have it set up. One of the things that's come out of this, and there's actually an NIA symposium on it in 2 weeks, is that most cases of autism have a similar hyperelectrical EI imbalance as Alzheimer's. And amazingly, when you do your screen for drugs, some of the drugs that work in one will work in the other. It's actually the basis of a clinical trial that's just started. And so these phenotypic screens are very, very important. We find things that really relate to the disease. And, we're glad to help people screen for drugs. We have something called the Reframe Library, which is every approved drug in every territory in the world, EMA, FDA, but Singapore, Japan, China, we have all the drugs. And also any drug that's made it through a phase 1 safety trial. It's about 14,000 compounds total. And so that's usually where we start our screens. It's a unique library that I'd like to offer to all of you that might want to do screening. So I would think that would be one point we could bring from our Southern California viewpoint to offer to our Northern colleagues.
Thanks, Stuart. And I also remember when we talked about these ideas that you were, and one of the things I'd like to address with you all is, how to best build, or how does the sharing occur? I mean, we saw from the pre-workshop survey that sharing occurs in all kinds of ways. And I think maybe the answer is there is no single way, obviously, how to share a resource. It really depends on the resource that is being shared. And I think, an actual collaboration, which is sort of what happened, which is what happened with the Genomics initiative, is there was a shared resource, and collaboration was actually the way how the resource was shared, as opposed to sending out things to various labs. So, but I think the point I'm trying to make is that there might not be, a simple answer, and it really depends on.
Well, and you've actually brought up my guilt about this. Sergei Pashka, you actually tremendously helped us. Some postdoc in your lab, helped one in my lab. You didn't get credit. You should be probably on my paper showing these electrophysiological phenotypes, because without your QC and quality control and input, we would have been, I think, lost. So thank you. But I think if we had a formal collaboration, this would be sped up so much more quickly.
Mm-hmm. Okay, great. So I think VG might be ready to hopefully her.
I can try if you can hear me.
Yes, I can.
Okay. So yeah, I was going to chime in, and what Stuart Lipton pointed out is something that I've been struggling with. So I'm a complete outsider to a field. I'm a neurophysiologist working on epilepsy, and I would like to adopt the IPS models because I do see the power of that, because there are patient populations with these rare genetic disease who have these complex mutations and multiple ion channels changing, both for a drug discovery pipeline and a mechanistic pipeline. It would be really nice to use iPS cells from these patients to get at some meaningful therapeutics for these patients. But coming from a completely different field, I see the barrier to entry, both in terms of the technology, and I'm not a molecular biologist or a stem cell physiologist. So having some kind of an interim place where, standardized models are, I don't even know if it can be standardized, because we're talking about neuronal circuits and we all want different types of neuronal circuits. And that's where I don't know whether it has to be individual collaborations with labs that actually do this, but they have to be local if you want to do functional assays. So maybe a distributed network of cores where there is the capability of generating some of these models, which other labs can adopt to use for their studies would be really helpful. Because protocols alone only get you so far. This seems like a really complex set of things to accomplish by a lab that is not fundamentally a stem cell biology, iPS cell biology lab.
And how would you ideally want to access, let's say if there was a resource lab that offers a model that you might be able to use to test your hypotheses, how would you want to access that model?
For me, it would be like the develop. So if we have the pipeline for getting the iPS cells and a local source for, say, differentiating this into different cell types that a particular lab might be interested in, that would be really helpful to get some of these systems into labs that want to use them.
Mm-hmm. Mm-hmm. Thank you.
But I think the know-how has to be somehow localized to different pockets. And I am in a pocket where there isn't much of neural stem cell work going on. So that's something that would be interesting to have across the SoCal region.
Mm-hmm. Mm-hmm. Yeah, thank you. Thank you for that. David Hausler. Mm-hmm. David, you're muted. So I think we haven't talked enough about stimulating the tech. What's going to make a difference over a period of years is how much you have improved the tech that creates these organoid models that are suitable for the science and then finally for the pharmaceutical development. It's time to really, I think, brainstorm about how you can do that. It's interesting, the Human Genome Project set aside some money to stimulate the tech. And of course, most of that tech stimulation came after the first draft was published. But if you've gone back, you realize that the price per base dropped by 1 million-fold because of tech development after that. You want to capture this, and you're not going to get it from companies that are trying to sell like an all-in-one system to you, because that price is going to be very high. It's a fast-moving target, a small, relatively small market. Well, I want to think about, I think you should think about it more like software, where there's an open source, highly stimulated community of engineers that are using 3D printing, microfluidics devices, Internet of Things, cell phone cameras, CMOS sensors, and so forth, putting together things and sharing those designs. And the best of those can emerge, but as flexible models, customizable models, not one protocol that one dictates that's supposed to work for everything, but a culture of creative engineering in a shared atmosphere that is stimulated by CIRM, because the actual, if you wait for companies to build you equipment that's exactly what you need, you'll be waiting forever, basically. We have to take the bull by the horns, stimulate a community of interaction, a community that has engineers interacting with CIRM researchers and building really, really cool stuff, not just software, but actual platforms. And I think what you're referring to is partly, sort of a opportunity for long distance experimentation, and not everything will be amenable to that, but
No, you know me with the long distance. I didn't go into that spiel. I'm just kidding. Yeah, maybe you can do your experiments in other people's labs, but that's too controversial. I'm just saying, let's share our tech. Let's co-develop our tech.
Mm-hmm. Okay, Ru, I'd like to hear your perspective on what we've heard, and on leveraging and collaborating, across institutions to make the best of all the things that we have.
Yeah, thanks, Uta. This has been really wonderfully stimulating for me to hear. I'm not a neurobiologist, and not in California either.
Mm-hmm.
So our take on this from the Allen Institute was, how do we make a resource that we made for our own work, which is cell biology, fundamental cell biology, as accessible to every lab around the world as possible, which is part of the, our philanthropist founder's mission, Paul Allen. And this has been a 6-year endeavor, and Bruce has been part of this journey with us. So it's nice to see him here. But I think someone mentioned here, the use of stem cells still is actually a privilege, right? Using it, the ease as well as the expense. Those are two things that we keep hearing are hurdles to using stem cells. Even though most people realize there are so many advantages, they are normal compared to cancer cells, all the differentiation capabilities. So those are two. And the third one, I don't think I heard here, but is the lack of maturity of a stem cell derived cell type. So that may be something fun where you can use innovation, could use bioengineering, you need a multidisciplinary approach. That's something that's missing. So let me come back to how we made our cell resource more available to everyone. We use two approaches. Mm-hmm. One, we do all the QC. We have 17 different assays we do for each of our lines that we do internally because we can and we have high standards. And then we employed a distributor so that they can distribute around the world. We don't want to be in the shipping business. Number two, we knew experts are needed for people who have never used stem cells, right? And I think, Vijay, you asked that question. So we went, as California was amazing. We have 7 stem cell cores around California who have experts. So they get the cells from us, they grow them, they bank them, and then they distribute vials as needed. So that reduces the cost for each PI. But they also, if you have a student or a postdoc who wants to be trained, they send them to the core and say, I want to learn how to make cardiomyocytes. I'll train you. And if you mess up, okay, the cost is not high. And once you realize in the first pilot experiments how useful those experiments are, then you can slowly invest. We would even give everybody a kit, like a media of, Matrigel, a bottle of Matrigel and a tube of vitronectin or whatever it was. So that's the hurdles for just kind of getting into it. If you want to expand the use of your resources, I think using your course, your major university course, is going to be really, really important because we've used them. Mm-hmm. Quality, making sure your resource is high quality, because as someone said, garbage in, garbage out, because you don't want someone to have bad experiments. I mean, experiments go wrong for so many reasons. So the way we picked our lines was based on ability to differentiate into many cell types, ability to grow and culture them for a while, genomic stability, and a bunch of other criteria. And third, we are very transparent. So we release all of our metadata, our genomic data. We start with 100 clones and release one. All of that information, we call it certificates of analysis, kind of like you get for chemical. So we are very, very transparent. We have a database, we share that. And that kind of makes people feel more comfortable to get into this. So I have lots more things I can share with you, but I'll stop there. And SOPs, we have video tutorials, so people can watch them, follow them, especially during the pandemic. Those were very useful. But I think getting more people to use stem cells is really going to have a big impact.
Ru, if you could also comment on your approach to taking the pain. I believe you have a way to take away the pain of of the legal stuff, the material transfer agreements, et cetera. Can you, which hasn't, I mean, it came up this morning, actually, Ralda Nimi mentioned it in her recorded talk, but I wanted to bring that out as well, sort of more of a practical issues with sharing.
Sure. So I'll give the short version because we could spend 3 hours on this. So we took a tiered approach. The first approach is what can we do so that nonprofit entities, like all the researchers out there, can have access to the cell line and the edited, CRISPR edited lines we make, right? So we use GFP, which gives us a lot of options, and we got the rights for that. So it's safe harbor. Then we wanted to make it available to commercial entities for research purposes. We still haven't gotten to the point where we can make it fully available to anyone. And that meant that we had to get the licenses and bundle them together so that we can then provide to commercial entities for research purposes with certain terms. But all of this is expensive. I'm not going to say this is not only hard to do, but very expensive. And we make these resources available to a fraction of the cost it took us to make this. So keep that in mind. This is a very expensive endeavor if you want to make this more open.
Yeah, thank you for that important part of thinking this through. David.
Yeah, thanks. There's a lot of good points being raised that I'm kind of, framing them in the sense of democratizing the process. And I think there can be a multi-pronged approach that addresses that. The technology development, I think, is really important, as David pointed out. And I think there's a way that you can do it. In the BRAIN initiative, we have a lot of technology development initiatives. But one of the key things in each of those initiatives, the way the BRAIN initiative is set up broadly, is that there needs to be dissemination built into the process. So somebody develops something, by the end of the project, they're giving it away. And they're telling people exactly how it's done. And in some cases, there are actually dissemination cores that are part of the process. And to the extent that those are relatively low cost of entry, that can be done relatively easily. For some of the approaches where there's a high cost of entry, and I'm thinking about things like robotic automation, and there's a relatively high cost of entry to the village culturing approaches as well. Those can be done through specialized cores that are placed here and there, but they're specifically tasked with serving the community. So I think there can be multiple approaches depending on the relative cost that it takes to do it, which will democratize the process broadly, depending on the needs of the particular investigator.
Yeah, thank you. I mean, your experience with implementing, shared resources, is really helpful. Same with Rue, that you have already, gone through many of these hurdles. Bruce, I'll see your hand is up. If you could also, whatever you wanted to comment, but then if you could pivot also a little bit to the question around the Prop 71 shared lab and how they might be integrated or be part of our next generation shared resources.
Well, yeah, I can address that right away. I think that the Prop 71 labs in fact, our laboratory at Gladstone was actually the first facility that was completed from using Prop 71 money, and we're still in that facility, and it's actually completely jam-packed full. So I think that's it's been tremendously helpful. I think that but we're at the other piece, though. I think that is that it's to make it so that it runs well, we really can't provide some of the services that we would like in terms of leading edge. We try to do as much in terms of some genome editing and things like that, but really, essentially, at this point, since we're not subsidized, it's relatively minimal services. And so expanding on that success, I think I'm sure other groups as well could benefit from that in California. So that's been good. And we've been servicing not just Gladstone, but just really the Mission Bay community in particular, because we're the facility at Mission Bay. So the other thing I was going to add is I was going to or second or third David Hausler's comment about the tech, and then make it a little bit more specific to some extent. One of the things, for instance, that we can now essentially make CRISPR cutting libraries of cells that have where every gene is knocked out, and then the challenge is essentially now for our particular experiment, we want to make a particular type of cell line, but that's a kind of resource. Once you actually have that machinery in a cell line, we've actually been able to provide those lines to other groups just to get started, say with CRISPR or other types of things. And that's tremendously, that's actually more costly than what we can do. We've sent out several hundred lines, and then also via the Allen Institute, via the wild type C, many more lines, and that's been great. But that's just that one line. But a library of cells which contain, say, a genetic modification of every gene, that actually is tremendously useful, because people can make cardiac myocytes, they can make neurons, they can make spinal cord cells, they can make all sorts of cell types with that, and they don't have to know much about molecular biology. They just need to be able to do the phenotyping. And so that's something, I could see a grand challenge of sorts where we have kind of whole genome perturbation libraries out of reference cell lines, but also in disease lines. And that being something that the individual lab doesn't have to know how to construct, but is actually focused on the phenotyping, because that's where there's the big barrier.
Mm-hmm. Yeah, thank you. You're sort of emulating a little what Aj also was saying in his talk this morning, about creating these resources of.
Yeah, Aj and I have been talking about this since he was at the Allen Institute, actually. So
gotcha.
Yeah.
Very good.
Yeah.
Dave Schaffer, if I could have you maybe could you make a few comments on shared labs, shared resources labs in the future, what connections you might see? You mentioned already that UC Berkeley's shared lab, which is called differently now, but is producing differentiated products, and so would be of interest, for this next generation of shared resources.
Sure. I'd be kind of curious. It'd be interesting to poll the 17, and maybe you've already done this, the shared facilities, to see how many are still in existence. I have a feeling that, a significant number of them are. There has been heavy investment in them historically by CIRM, and they, are probably close to being, fully functional again with fairly, non-onerous investment. And that could, serve at least at some campuses as the basis for a shared network. I will fourth or fifth the comment that Dave Hausler made, in that, as an engineer, obviously, we're very interested in developing technologies. We don't want to, and I agree with everybody else, standardize differentiation protocols. We don't want to stifle innovation. We don't want to miss biology that, could be missed if we were only using one protocol, et cetera. And in addition, everybody's working on different problems. But I'll just offer that there are, we've been working on, and others have as well, pretty low barrier of entry technologies that can be used to create much more uniform, more reproducible differentiation systems. So, for example, Eileen mentioned earlier that, as you move from 2D into 3D, or you move from in vitro into tissues, there are properties that are just, present in tissues that are missed in culture models. Mechanical properties, elastic stiffness, viscosity, the capacity to pattern differentiation, pattern vasculature. So we have been working with really simple, we're not interested in making complicated systems that only one lab can use. These are intended to be low barrier to entry. Material systems, optogenetic systems, where you can get very precise patterning and very reproducible systems. So we're getting like the same pattern in an organoid in 90% of the organoids within a culture. for several different stages of development, from gastroloid through neural tube. So, and we'd be happy to share those resources.
Mm-hmm. Yeah, thanks, Dave. And Eileen has some more comments along those lines, I suspect. You're muted.
You might be on mute.
Thank you. So just sort of in that vein, the last few comments combined, I think one of the challenges that comes out is that training is still incredibly important. Bringing new people that are, going to be moving into working in iPSCs or, either 2D or 3D or more complicated organ tissue engineering types of models is going to be critical, like making that pathway for folks to be able to do that, making a pathway for people whose specialty might be in bioengineering or gizmos to be able to apply what they do into the stem cell space and really make advances is important, right? And so how do we bring that together? In the context of the previous version of the CIRM cores, I wonder if we might think about funding something that's like, a resource of staff and basically hotel space that could be the glide path in order to make that happen, right? To just work on lowering some of these barriers and open up what the path could be to access all of the different resources that are at the different sites around California. And you can think about a lot of ways that that could come into play from a financial perspective that could be workable.
Mm-hmm. Okay, thank you. So we are about 20 minutes out from the end of this discussion. And I think what I have not achieved is to get an answer to the question, is it worthwhile for CIRM to pursue a network of shared resources labs that would be focused on stem cell-based modeling? And I think the answer, if I can just put it in this, I think part of the issue is that there are so many issues or so many things to consider. And ultimately we'll have to, if we want to go forward with something along these lines, we'll have to focus somewhere, at least initially, if you want to, think about establishing shared resources as they relate to cell-based modeling. I don't know if anybody specifically wants to comment on that very big question, and I appreciate that it's what we've talked about is very complex, and there's all these different things to consider, and we couldn't build possibly everything that has been suggested, and somehow we need to figure out, or we will figure out how to go forward. I don't know if I at this point could get somebody's just brief comment on that. And then I really do want to spend the last 15 minutes or so making a connection to tomorrow's discussion, because I think, as many of you have already alluded, there is a connection, between the biology that could be created with shared resources like the ones we've talked about, but how can we make most of what comes out of all this research? And that's what we want to explore tomorrow with our session on the data infrastructure. And I do want to spend a few minutes getting feedback from you all, not all of you, simply because there's too many of you, on what it is that experimentalists are looking for when thinking about collaborative data sharing and collaborative data analysis. So I'll have Ilyas and Bruce make some comments before we then go into that last portion of today's discussion. Ilyas.
As you pointed out, very complex questions, and many of the things we're hearing, they're not mutually exclusive, right? So it can be really a focused effort to generate new knowledge, new tech, new assays, etc., versus therapeutic development, right? So what do you want to translate? What do you want to achieve in terms of helping patients, right? I mean, this is a different approach, in my opinion, and this could be, again, it's not mutually exclusive, but it can be predefined as a specific goal, and then really having a more targeted discussion around, so what needs to be done to, develop a drug? Is the phenotype going to be druggable, right? Those kind of questions can be, I think, discussed and defined, and then based on that, you take action. Yeah, these are my thoughts around that.
Thank you. Thank you. Bruce.
Yeah, just quickly, I think most people would say that it's worth it. But I think it's a really good question about what would the landscape look like without sort of shared resources, and then what would the shared resources do that would empower the other portion of the portfolio. And so if you imagine individual kind of hypothesis-driven labs and labs focused on different diseases and so on, how would they be more powerful, essentially, in terms of getting what they do done, what they want to do done, because of shared resources that they don't have to build in their own labs? And I think I think it's pretty obvious that that's going to be the case, but I think it's easy to sort of break it down to, you have sort of the power users who would be sometimes the ones who are making the resources, and they can actually make better technology, as it were. But then there's also, I think, the sort of the lowering the entry to the barrier to entry for the people who are not doing IPS research. And that's really a community that I think is important to get in, because many of the researchers here were not, that are on the line now were not stem cell researchers, when Prop 71 happened, and a lot of people came on during that time because of these resources. And so that's the thing that I'd like to just think about, how you have the sort of the power user one, and then also the people who are the ones who are not doing IPS research but would want to do something in human cells, who are really interested in therapeutics, and that this is a great way for the CIRM to actually bring more people to the table, increase the size of the pie, particularly in the California researchers. So that's where I would summarize.
Yeah, that's a nice way of looking at it. Mike.
Yeah, just a quick comment. I assume you're going to have similar conversations with other vested groups like cardiovascular, maybe liver, whatever. And so I imagine then you'll want to, especially for tomorrow's discussion, make sure that the control lines can obviously be shared across all these groups, and the fields and things, the data collection types of things should also be homogenized. So putting this in a bigger picture.
Yeah, thank you, Mike. And while I have you, maybe you could start us off. Actually, Jackie, why don't I go to Jackie next?
I'm just going to underscore many of the things that were said previously. I think as someone who is relatively new to the field and trying to think of, as I listen to the discussion, I'm wondering what you and the other leaders at CIRM are, who is your target audience? If you are in fact trying to bring in people who otherwise have a high barrier to get started in this, you've already provided the cellular resources, but what do you do now? You go to Fuji and you buy a frozen vial of cells. And that is, I think, too challenging for the uninitiated. And so to provide a resource that can jumpstart individuals, as has been discussed, I think would be really a tremendous boon to the field. I think one of the things that's sort of been a stall to the liver field is that many of the cells die out at the endoderm stage, and it's very challenging to get any or less than just a small fraction of the lines all the way to differentiated cells. And so, being able to tackle that has, or being able to identify methodologies that can improve that would be a great resource. But that's a very different goal than, also, supporting innovation. And so I think that there is room for both. And it's just, I would love to see more democratization of the process, but I think not at the expense of the super users and the innovators.
Thank you. Ash. Are you speaking? If you are, we can't hear you.
Oops, sorry, my mic was off. I think it's super interesting to hear kind of the different perspectives on innovation. And it was pretty interesting to think about, there's kind of coming from Sergio and Arnold in terms of innovation, in terms of the systems, making more complex systems, 3D models, kind of innovating on the protocol development, which I think is a really, interesting area to go, and I think is very important. I think on the other side, kind of innovating in terms of phenotyping and kind of innovating in the way that Steve Finkbinder is mentioning, and the way that InSitro is trying to do in terms of having a large standardized collection and then using machine learning to identify commonalities. I think there are just different ways to innovate, and I think we need to kind of consider those different approaches in terms of how we move in terms of generating these resources. I think both are important, but I think they're just different. There are different ways to innovate, kind of across those different areas.
Mm-hmm. Yeah, thank you. No, it's very exciting to just listen to your ideas and all the work that's already going on. Okay, to pivot in the last few minutes to the needs and desires of experimentalists in terms of collaborative data sharing and data analysis. David Panchisian, if you don't mind, I'd like to call on you, since you are, you live in both worlds, and if you could make a few comments on your perspective on what it is that experimentalists wish they had when it comes to data sharing and made easier and data analysis, collaborative data analysis across laboratories made easier. Is this something you could comment on?
Yeah, sure. And I've sort of come upon this the hard way. I think one of the biggest challenges is for investigators to retrieve data in a way that's understandable and usable. And a lot of that has to do with the way data is formatted, metadata that's associated or not associated with it, and differences in the way it's stored in different archives, and whether they do or do not communicate with each other. And so I think, and on top of that, the ability for the data to be structured and shareable in a way that data scientists and computational scientists can actually use it. And so I think there needs to be sort of a handshake relationship between the people who are using the data and the people who are managing the data. That's the biggest challenge, and it's a hard problem. And I know that we're going to go into that in a lot of detail tomorrow, but I think that's sort of the overarching challenge that needs to be dealt with.
Mm-hmm. Thank you. Aparna, if I actually can just call on you, you're an experimentalist who, at least according to Max Häusler, is very capable of data, handling data as well. And, and your, and some of your other colleagues, facility with, again, living really in both worlds. I'm curious if you could talk a little bit about what your dream would be in terms of a broad access to data.
Sure. I think that having collaborated with a lot of people who are interested in the biology but need the data to motivate that biology, I think that having data to be accessed in multiple ways. So one, having the raw data for the people who want to devise new algorithms, who want to interact with that data, who want to be able to get it in its rawest form with all of the complex metadata. I think that that's essentially important. But then also having some community standards for when we are doing something as part of a CIRM consortium or something similar, how do we process that into a uniform way that someone who is not an expert at all of those steps that take days, weeks, and months to get to a counts matrix, how can we get to some processed form of the data that can be interacted with and analyzed quickly to ask a biological question? So one of the examples that Max has really enabled with his browser is if you just want to know what cell type a particular gene is expressed in across any of his 100 data sets, you just go to the browser and type it in. You don't need to know how to make a feature plot. You don't need to know how to make a violin plot. You don't even need to know how to open R. And I think that that level of accessibility has really enabled me to have a lot of good data conversations with people who are more biology focused, but then also to interact with the data at its, in its rawer forms as we want to devise new tools or analytical methods. And so I think having both paths is really important for the field and for accessibility.
Mm-hmm. Thank you for that. Yeah, I mean, I think, one realization is, of course, that many biologists who are growing up now, also have to be data scientists, and that will help, with, bringing these collaborative platforms to fruition. Steve Finkbeiner, maybe can you also comment on your perspective on data sharing and collaborative analysis?
Yeah, I mean, I'd say that we have benefited tremendously by being part of consortia and being able to structure portals to be able to share data. I don't know if Clive's still on the call, but he's one of the members, for example, of our Answer ALS consortium, where this has been a big focus. And I think being able to both have a place where the data can reside, and even a place that can even house some of the computational tools that users might want to take advantage of, so that the compute and data are in the same place, can really make it very powerful and something that people will make tremendous use of. And I would just underscore again my own opinion that having a lab resource network, specifically a network where we can aggregate data, really would enable us potentially to achieve a scope and scale of data that really can enable things that really aren't possible by single labs, in terms of particularly using deep learning approaches and others to be able to get insights from those data that are difficult when the data sets are smaller. So I think this is very valuable. It's a challenge to do all the harmonization and get all the metadata in order. So it's not for the faint of heart, but I think there's a lot of rewards for people who where you can get it to work.
I'll just follow up to that.
Yeah, please.
One of the challenges with Answer ALS was metadata, was accumulating all of that patient data and having it available at the portal. But the really cool thing now is if you see something in the data you pull off the portal, you can go and get that exact line and have it in your lab within about a week and rerun an assay or do something. I think that tying together, along with its genome, along with its clinical data, and that resource, I think, we just done it for ALS, is something that I think would be very valuable to the field, where you can just pick a line, it's all annotated, genome sequencing is there, the clinical data is there, deep clinical data, and now you can run your experiment, whatever your organoid experiment is, with all of that behind it. And then when you publish, it's kind of like, oh, that was the line that we all know about, and now we've discovered this.
Yeah, and just to David's point about innovation, I know most of the, thinking is around actually innovating around the production of the models, but there's tremendous innovation possible, I think, around the analytical tools too. And I think if you have a central place with data, that certainly would facilitate it. And I think there would be even opportunities to capture some of that innovation, in the cloud, to be able to disseminate to others as well.
Yeah, thank you. Mike, what's your take?
Well, so I assume we'll talk about all these things tomorrow. But the metadata is the hardest thing to get people to enter and fill out all the fields on. So you really need to think of carrots and sticks to make it all happen.
Gotcha. Yeah.
I'm sure we'll talk about that tomorrow.
Yeah. Yeah.
And I agree with everything else said earlier.
It would be great if there was an experimentalist who wanted to chime in about sort of the hurdles they see or why. I mean, we heard already a little, Mike just said, entering the metadata. I can totally see that being like a huge hurdle.
Well, to be honest, most experimentalists don't really budget for it. For entering data, it's always an afterthought. And even today, that's still the case for many folks. I think Power Labs kind of know to build that into the budget and such. They have a data handler. But, these one-off labs who want to do these fun experiments, it's the last thing they think of is submitting the data, getting it well annotated.
Right.
So I think somehow getting that in the mindset early is very, very important, and probably in any RFA sorts of things that go out that might use these or might be contributing data, probably should be right in there. That data sharing and budgeting appropriately is critical.
Mm-hmm. And that it'll be monitored.
Yeah, actually NIH has, you probably know, has a data resource sharing plan that everybody has to fill out. Although these days it's kind of lip service, so I'm not sure.
Yeah, that's what I sort of meant. It's, going from lip service to actually making it happen. Yeah.
Depends on how much of a bad cop you want to be.
Yeah, we used a practical solution now. When you design something right from design, it's a drop-down menu. So we work with our software engineers, right? So we are very multidisciplinary. And to even freeze down a vial, when they go to the LN2, if the metadata is not in, you cannot freeze your vial. So people got so frustrated now. So we trained.
Yeah.
So if you set up from the beginning, I mean, we learned the wrong way for 2 years. We didn't have anything. Then we made it like barriers. If you don't have metadata, you can't go to the next step, and then it has to be searchable by someone else. Those are the two criteria.
Gotcha. Yeah. Thank you for that. Yeah.
I'm lucky to be at a place where it was, kind of started by machine learning engineers and software engineers. So we were forced to have to enter everything in the way that could be utilized downstream. So it's actually been quite nice, but it does take a big change in the way that you do things. And the way that a wet lab biologist would do things, kind of is very different than what a software engineer or what a, kind of machine learning engineer wants out the back end.
All right. Thank you all for all these amazing comments. Luckily, I didn't have to write all of this down today. We will dutifully summarize the deliberations today and take into account what we've heard. And before I hand it over to Rosa, I will call on Ilyas.
Hi again. So just wanted to put out, we just finished a manuscript and put the preprint on bioRxives. So it touches on the issue of democratizing data analysis. As you pointed out, so biologists oftentimes generate the data, but they are really depending on bioinformaticians. So people with special expertise who can analyze the data and get back to the experimentalist. But in this case, this resource we have built, so we're calling this CIRM, allows rapid and reproducible RNA-seq data analysis. So this is bulk RNA-seq as well as single-cell RNA-seq. So in a more reproducible and standardized and more scalable way. So biologists can really interrogate their data. They can quickly make decisions on redesigning experiments, redoing experiments, etc. So it really shortens the timeline of data generation to data analysis and then deciding on next steps. So yeah, this is just as of today available on bioRxives. So we would like to hear feedback on how this might be helping the field. And part of this resource is also an application that is integrated, and we call it the iPSC profiler. So it helps with measuring pluripotency and differentiation into the three primary germ layers. So yeah, so in addition to what we are typically using like pluritest and scorecard, this would be an additional useful tool for the community. So yeah, look forward to receiving feedback on how this may work out in other labs and yeah, thank you.
Yeah. And thank you. And then also underscoring, the ease of use, for the non-expert, non-data expert, and keeping those things in mind. So I'll end today's discussion. And I'll hand it over to Rosa in a moment. I do want to just thank you all for being here today. Also for each one of you talking with Rosa and myself prior to this meeting. We have lots of notes from those discussions as well. They didn't all come up during today's discussion, but will also be considered as we think about how to move forward. Thank you all. This was fabulous. Rosa.
Thank you, Uta, for moderating such a great discussion. It was very informative. I think I was going to summarize all what we heard today, but I think it was going to take quite a bit of time. But the goal this afternoon, I think we had a great discussion. If CIRM was to support the creation of shared resource labs for stem cell-based modeling, whether that could be feasible. And we heard a lot about the hurdles and what it depends on. And many of the things that we heard is that it depends on the ultimate goal of these models. There are also many resources that already exist, and we need to learn how are we going to bring them into the fold. There was a great discussion about the types of disease models, and I think a particular comment that I think is important regarding the standardization of protocols and stifling innovation. We agree definitely that we don't want to stifle innovation. But we've heard over the past 2 years from expert stakeholders input, as well as tomorrow we will see it in the results of the registered attendees that completed the survey. The field needs some lines and protocols that can provide the firm foundation and a reference for generating reproducible results and to provide results and access points for non-experts. So innovation will definitely continue, and particularly in the specialty labs. But this is something that we will need to pressure test more. And then one of the points I wanted to make is I think in terms of the data sharing, and I want to link to tomorrow's discussion, in terms of data sharing and collaborative analysis, there's a lot to talk about. We heard from David Pankinson about interoperability and the handshake between data generators and data managers. And I think that this is going to be provided tomorrow. The discussion will focus on what could be the role of a knowledge platform and how could we develop, and if CIRM could have a role in the development of a data infrastructure, a data management center, and the challenges to do that. Processing data into a uniform way that can be interacted with to ask biological questions. For example, the cell browser is something that a knowledge platform could be allowing us to do. A knowledge platform, as you know, and you will hear tomorrow, is a cloud-based infrastructure and software ecosystem that will provide tools and applications and workflows to allow collaborative shared analysis in a computing environment with secure data access and security protocols. And this is something that we will talk about. I also heard a lot about the metadata. It came up in different points, and we will talk about metadata tomorrow. And I think what I would like to say is thank you, and I'm looking forward to tomorrow's discussion. We will learn a lot about the data platforms, and we will hear a lot about the infrastructure principles and capabilities, as well as excellent examples of collaboration in the cloud and data use oversight system platforms, and how this is applied to specific cases. And I hope that everybody will join us tomorrow in that discussion. And I hope that you all have a great rest of the day. Thank you all.
Thank you.